Good morning, and thank you for joining us in the third edition of the Latin American Ocean Acidification Network LAOCA webinar series. My name is Fernando Mejias. I am the Communications Director of the Coastal Socio-ecological Millennium Institute, SECOS, which together with the Millennium Institute of Oceanography, IMO, participates as co-sponsor of this series of LAOCA webinars. We also thanks to the project REDES 180012. I therefore welcome you to the LAOCA webinar, Ocean Acidification and Climate Change, Perspectives from Evolutionary Ecology. For all of those who are joining us for the first time through the LAOCA YouTube channel, as I pointed out before, LAOCA it's the Latin American Ocean Acidification Network. It was born in December 2015 in Concepcion, Chile, with the mission of becoming a regional node of articulation, communication, and strengthening of the understanding of the ocean acidification process in Latin America, and its interactions with other local processes, as well as its implications on marine ecosystems and their ecosystem services. LAOCA is currently integrated by more than 70 members from countries such as Argentina, Brazil, Colombia, Costa Rica, Cuba, Ecuador, Peru, Panama, Mexico, Chile, among others, and surely many of them will be connected and seeing us right now. We salute them all. During all these years, LAOCA has played an important role in coordinating and articulating all the efforts related to increasing the capacity to observe the chemical changes that are occurring in the oceans. Also, as well as in the evaluation of the impacts that these changes have on the populations, communities, and diverse marine ecosystems that characterize this part of our planet. Finally, LAOCA seeks to become a network that can collaboratively transmit the progress of research on this topic to decision makers, private organizations, the society, and others interested in information related to the study of ocean acidification in Latin America. LAOCA currently coordinates its activities thanks to the work of an executive committee that during the period 2021-2023 is being co-directed by Dr. Carla Berghoff from the National Institute of Fisheries Research and Development, INIDEP, in Argentina, and Dr. Cristian Vargas, academic of the Faculty of Environmental Sciences of the University of Concepcion in Chile, and researcher of the Coastal Socio-ecological Millennium Institute, SECOS, and the Millennium Institute of Oceanography, IMO, who will be introducing our guests today. Dr. Vargas.
Okay, thank you, Fernando, for the presentation. So, hey, everyone, it's a real pleasure being able to join you virtually today. This is the third LAOCA webinar, which is taking place within the framework of our international LAOCA course entitled Ocean Acidification and Other Stressors Associated with Climate Change, a look from evolutionary ecology, which is carried out from Monday, October 25, and ending the next Monday, November 1. So within the framework of this course, we have thought to organize this special webinar where not only the selected participant to this course may be able to attend it, but also can be broadcasted through our LAOCA YouTube channel to the entire Latin American community and of course the rest of the world. Today we have three great speakers, and by virtue of time, I would like to introduce our first guest, which is already connected. Dr. Sinead Collins, welcome to our LAOCA webinar. Thank you very much. Sinead is a researcher from the Institute of Evolutionary Biology at the University of Edinburgh in Scotland. Her lab group uses photosynthetic microbes to expand evolutionary theory with the aim of predicting phenotypic evolution in changing environments. This includes projects that look at the role of fluctuating or complex environments in the action of natural selections, the links between trait plasticity and trait evolution, and the possible role of epigenetic on evolutionary outcomes. Concretely, Sinead and her group are interested in making generalizable evolutionary predictions about changes in fitness and phenotype from the contemporary characters of populations and their environment. So welcome, Sinead. Thank you for taking time to join us. Sinead will share with us a great presentation today entitled Multi-Trait Evolution, How Many Ways Are There to Be a Diatom? Thank you.
All right. Thank you so much for that introduction. I'm going to now attempt to share my screen. So that's the first bit. And can someone give me a thumbs up if you can see that presentation properly?
It's perfect. Thanks.
Perfect. Excellent. So thank you for that wonderful introduction, and thank you so much for having me here today. I'm so pleased that this webinar is able to happen now that we've all become really used to Zoom. So today I'm going to be talking about how many different ways there are to be a diatom, and I'm going to be presenting new work. So I'm going to leave you with more questions than answers. Hopefully that's okay. Now, I tend to think of what I do as projecting changes in life-giving slime in aquatic systems, and this is because phytoplankton, the organisms I work on, very much are the lungs of the planet. Ocean ecology and food webs depend on them, as do we for about half of the oxygen that we breathe. I'm not presenting work that I've done all by myself. I'm presenting work that I've done in collaboration with two other labs, that of Naomi Levine in Los Angeles and Martina Doblin in Sydney. And Jana Hinners, Nathan Walworth, and Phoebe Argyle are the three fearless postdocs that have worked with all three of us for the past 3 or 4 years to do this project. So, think of us as team phytoplankton. Now, if you're on team phytoplankton and you're interested in ocean global change biology, and this is what you're dealing with. You're dealing with phytoplankton, which are a really highly diverse group of organisms, and they live everywhere that there is water. They play a major role in regulating key climate drivers, such as the carbon cycle, and they also underlie things like wild fisheries, as well as subsistence fisheries, actually. And they're responding to really complex environmental change. They're not just dealing with ocean acidification. They're also dealing with warming and changes in light regimes and changes in nutrient fluctuation, and actually changes in the grazers that regulate them from the top down. And the most striking thing about being on team phytoplankton, if you're me, is that the characters of phytoplankton are very poorly constrained in Earth system and ecosystem models that we use to predict future climate and future ecosystems. So sometimes I really feel like I'm dealing with this monster here. This is the Austriokraken. It's what the world's tiniest picoplankton would look like if it was attacking a giant ship. So the question we're asking is, how are you going to be a phytoplankton? How do we constrain the number of different ways there are to be, in this case, a diatom? Well, we actually have a really great start from modelers because phytoplankton are represented in ecological and biogeochemical models. So the way you be a diatom is, first of all, you have traits. Traits can be anything: cell size, photosynthetic rate, nutrient uptake rates, sinking rates, grazability, you name it. But you have traits that are correlated both to each other and connected to ecological and biogeochemical functions. So connected to things like how likely you are to be grazed or how much carbon you sink. This tells us which traits we're interested in, right? And an example of the way we get interested in these traits is by saying, here's our model. Phytoplankton, it takes up carbon, phosphorus, and nitrogen with some quotas, and then they have some internal metabolism that deals with that. All of these processes are modulated by light and temperature and tend to scale with cell size. So through models that tell us about the function of phytoplankton, we get a list of traits that we care about. Of course, we care about all traits, but as people who work on anything global change related know, we're on a bit of a timer here. So we're interested in traits that tell us about a certain type of function. Now, we also know what types of evolutionary constraints we need to know about through this laundry list of traits. And constraints are things like trade-offs between traits. Now, this is just an example of a trade-off where we are looking at the irradiance niche, so the amount of light that an organism uses versus the amount of nitrate they need. And you can see that if you live at high light, you also live at low nitrate. And this sort of trade-off or relationship constrains the combinations of traits that can occur. And most importantly, you can have all of trait space here. So all of these values of nitrate are possible, and all of these values of irradiance niche are possible, but only a subset of them are energetically feasible, and only a subset of those actually occur. Now, if we can actually constrain or guess at, in an educated way, where this pink circle is, then we can predict where and how much phytoplankton happen. And that tells us things about primary production and biogeochemical cycles. Now, the smaller we can make this red circle, the more accurately we can guess the constraint, the better our projections will be. You don't want to be using all of trade space. You don't want to be using all of the energetically feasible trade space. You actually want to be only using the occupied trade space. The problem is there are a lot of traits. So I've just listed here some of my favorite traits, and you have nutrient uptake kinetics for all the different nutrients, or at least for nitrogen, phosphorus, and silica if you're a diatom. You have cell shape and size, photosynthetic traits, cell composition, sinking rates, temperature sensitivity, and then something I'm not going to touch on today, and that's often ignored, life history. So it's just too many traits and too many correlations if we treat them 2 by 2. And that's our second monster, the eye spot of Sauron, for those of you who are Lord of the Rings fans. And here's some tiny viruses attacking it. Sorry, sometimes in my lab when we get a little overwhelmed, we make phytomonsters. So diatoms are quite complicated, and I didn't start off studying diatoms. So at this point, when we started thinking about how do we think about all of these traits co-evolving at once, and how do we think about a trade-off that's not just between 2 traits, but between n traits, so maybe 6, 8, or 10 traits, I immediately thought, bring me back to Chlamydomonas. Chlamydomonas is a freshwater model system. It's sort of a green yeast. It's a lab model system that doesn't, where we don't know a lot about what it does in the real world, but it's really tractable in the lab. So it's a good model system for experimental evolution. And while it's not marine, so far, all of the basic things that we figured out in Chlamydomonas have held true when we checked them in marine phytoplankton. So this brought me back to an interesting paper that I had reviewed in 2014 and was published in 2015, and then I forgot about for about 3 years, but it stayed in the back of my mind as something that was really cool, and I could never figure out why it was really cool, or at least it took me 3 years to do so, which was Malcolm et al. Took 18 genotypes of this freshwater model system, Chlamydomonas, and they grew it in 30 different environments. Wow. So this is a terrible way to look at data. What you're looking at here is a bunch of different environments and growth rate. And then on the x-axis are all the different genotypes. Now, if you look at the data like this, all you can really get out of it is that there's variation in growth both within environments between genotypes and between genotypes within environments. But we already knew that. You don't need a giant experiment to tell you this. What was really cool about the Malcolm experiment was that they said, why don't we look at all of these traits at once? So for those of you who are not familiar with principal component analysis, it's going to be a big part of this talk. So briefly, principal component analysis is a way of reducing the dimensionality of a data set. Usually you have as many traits as you want. In their case, they had 30. In our case, we'll have 8 or 9 or 10. It reduces this into something with fewer dimensions. Usually we look at the 2 principal components. It makes new variables that are linear functions of the original variables, so the original traits. No information is lost when you're making a principal components analysis. You can reverse it and get your original data set back out. You maximize variance along successive axes. So it's a statistical technique that tells you about the structure of your underlying data set. It tells you which combinations of traits change in which way to get the most variability in your data set. And the important thing about this is that it emphasizes relationships between variables. And in our case, variables are trait values. So the Malcolm et al. Took their 18 accessions and 30 environments and made a PCA, which explained about 50% of the total variance, which is pretty impressive. The neat thing about this is because it works so well, it supports the idea that even though there's lots of genetic variation, the responses to environmental change are very constrained. They occur along a few major axes, and this should constrain what evolution is likely to do as well. And this means we might be able to constrain how many ways there are to be Chlamydomonas, or how many ways there are to be a diatom. It does rely on one key assumption, which is that the trait correlations themselves are constrained, so that trait correlations don't evolve much. So what we're asking is that a reasonable thing to think about? If we assume that this trade-off remains constant over time, at least within one taxa, so one species or one genus. Well, we happen to know that's not true. If we look at data from an evolution experiment done in my lab, again with Chlamydomonas, where we took different genotypes of Chlamydomonas and evolved them at high CO2 for 900 generations, the outcome isn't super important here in terms of what traits changed. What's important is that the trait correlations evolved a lot. So we looked at the study looked at many different traits, but we took a subset of 4 traits here. And we looked at the ancestral correlation and the evolved correlation. You can see that between traits 1 and 2, there was originally no correlation, but after 900 generations, there was a correlation of 0.6, which is very strong. Perhaps more striking is between these 2 traits, the ancestors had a correlation of minus 0.9, so a 90%, well, an R-squared of 0.9, which is insane. And by the end of the evolution experiment, those 2 traits were completely uncorrelated. So we know that trait correlations evolve readily and quickly. We thought about how to model this. So this is Nate Walworth, who bravely did all of these models. And we thought, okay, well, if we're dealing with 4 traits instead of 2, how can we model evolution if more than 2 traits matter for fitness? And the traits we were dealing with Chlamydomonas were not trivial ones. They were things like cell size, reactive oxygen metabolism, and photosynthetic rates. So they're traits that have a clear fitness value and would affect the ecological function of Chlamydomonas. We thought, okay, well, let's evolve them on a principal components landscape instead of a more traditional fitness landscape, which would just have 2 traits and fitness as the y-axis. The important thing to note here is because we did the experiment in the lab, we know what the starting point is and we know what the endpoints are. So we know that the evolutionary trajectories that we're modeling are reasonable ones. And that's just showing these are the ancestral correlations, the evolved correlations, and we also knew the ancestral and evolved trait values for each one of those traits. What I'm showing you here is the actual simulation itself, and I'm happy to go through the details of that with someone later, but it's just an adaptive walk of the type that R.A. Fisher came up with. And the fitness peak that we got experimentally is this red dot. The ancestral point that we started with experimentally is this yellow dot. And here we see the evolving population. You can see that by the 10th step in the simulation, it's approaching the peak. And by the 2000th step, it's at the peak. And so this shows that our simulation worked. The populations evolved. Interestingly, even though they all ended up on the same peak in PCA space, they had 4 distinct phenotypes. So that's a way to say there are multiple ways to occupy the same dot in PCA space in a traitscape. So what we did was we looked at how much of that cryptic variation or those equivalent phenotypes in terms of PCA space we could produce based on how much we let trait correlations evolve. If we assume that trait correlations don't evolve, so we just take the ancestral traits and fix them, then we find out that we fail to produce the 4 populations in most cases. So we do get evolution in this model where we don't let trait correlations evolve, but we don't get as many outcomes as we expect. And so the diversity of outcomes is lower. Whereas if we let the trait correlations evolve, we get the diversity of outcomes that we actually observed in the experiment. Why am I telling you all this? Well, I'm telling you all this because when we model trait evolution in almost any ecosystem model, we assume that trait correlations and trade-offs can't and don't evolve. What our model shows when we combine it with an experiment is that you only get the right number of outcomes or a reasonable number of outcomes when trait correlations are allowed to evolve. And not just allowed to evolve, but allowed to evolve rapidly and dramatically. The other interesting thing is that you always get a number of cryptic phenotypes underlying the evolved phenotypes in principal component space. This is to be expected based on the way that PCAs are put together, but it's still really interesting because it does tell us that there are multiple ways to exist in a single point in PCA space. And that's going to be important when we start thinking about how to represent multi-trait phenotypes and how they evolve. So that's all well and fine, but it was a simulation based on an experiment that was never meant to answer this question, and that was not done with a marine diatom. So enter the marine diatoms and the postdocs that love them. So we did some evolution experiments and physiology experiments with between 6 and 12 different genotypes of Thalassiosira. Thalassiosira is a lovely diatom. One of the reasons it's lovely is because it has so many genotypes represented in culture collections, so that we're able to, in our study, go across the ranges of things like cell size and growth rates and, to some degree, thermal niche width that one would expect in Thalassiosira. The two postdocs that worked on this are Jana Hinners, she worked with me in Edinburgh, and Phoebe Argyle, who worked with Martina Doblin in Sydney. The basic question we asked was, if we let diatoms evolve, will all of the evolved lineages be on this line? Will they all maintain their ancestral trait correlations and evolve primarily by sliding up and down this line? Or do we expect to find populations that evolve out here as well? And the reason that's super important and interesting to us is it asks the question, or answers the question, of whether we expect evolution to produce fundamentally new types of diatoms as oceans change, or just add more points to existing patterns of variation. So if you sample enough now, you will have sampled all the ways there are to be a diatom. And evolution won't come up with more, it will just shift around what it already has. So if we're looking at this in terms of traitscapes, we're asking how constrained is movement on trait spaces, on trait landscapes. So if we think of our principal component space here, PC1 and PC2, there are three options. You can have this case of isolated peaks, where you have an ancestor in yellow. You then push it off its peak, and I'll go into how we do that, but you shove it off the fitness peak in a random direction, and then you let it re-evolve. And when it re-evolves, it just goes back to the same place. That would tell us that that peak is relatively isolated. It doesn't mean you can't escape it. It means that escaping it requires going through a fitness valley that's extremely low. And so the population would be likely to go extinct if it tried to escape that peak. We can have a case of connected peaks where we have our ancestral population, we shove it off the fitness peak in a random direction in red, and then it makes its way to a different fitness peak, but the fitness peak is one that was occupied by a previous ancestor. So this is a case of it sticks with pre-existing variation, even if it doesn't stick with its own fitness peak. Finally, we can have an ancestor that shoved off the fitness peak in a random direction, and then when it re-evolves, finds a novel peak that no ancestor had previously occupied. And that's a case of innovation. And this is where the talk gets a little weird. This is Jana Hinner's paper, and this is still an analysis and rather heated discussion between everyone involved in the project. This is how you do a perturbation analysis like this. We looked at 9 traits. Obviously, looking at 9 traits and all interactions between 9 traits, including 3 and 4 and 5 and 6 and 7-way interactions, is a terrible idea. That's why we've been talking about these multi-trait landscapes. What we did was we had 6 ancestors for the evolution experiment. We had 6 replicates of each ancestor, so these are 6 genotypes with 6 replicates. We then reduced selection for up to 9 bottleneck transfers, and the way you do that is that you transfer very few cells in batch culture, and this means that chance is stronger than natural selection during this time. And what this does is shoves those populations off the fitness peak in a random direction. The populations still have to be viable, but their fitness can drop, and it does. Then we let the populations re-evolve under 2 different sets of conditions, one where they re-evolved in the ancestral environment, and one where they evolved in a warm environment, 24 degrees. 24 degrees is not stressful for these diatoms. It is still below their thermal optima. All right, let's start. What I'm showing you here are the 6 different genotypes and the traitscape difference. So the Euclidean difference in PCA space where that they went from their own ancestor. The ancestors are in black, and the populations that re-evolved at 20 degrees are in blue. The red are the populations when they were shoved off the fitness optimum. And the important thing here is you can see the blue populations don't differ from their ancestors. They occupy the same point in trait space. So they're forming these isolated peaks. In contrast, populations that re-evolved in warmed environments, the green ones, sometimes found a novel or connected peak. They did sometimes also go back to their ancestor. So in a new environment, all 3 outcomes are possible. So generally the bottlenecking seems to have worked. We can tell this because the red boxes are very large, showing that variance increased dramatically during the reduced selection treatment. Strains return back to the ancestral bit of trait space when they re-evolve in the ancestral environment, but they can move around trait space when they evolve in a new environment. So even though they're moving around trait space, we see that they evolve new trait values. And this is an example of those cryptic phenotypes I showed you. So here are two traits. They're just representative traits. We measured 9. Here are 2, cell size and chlorophyll A. Along the bottom are the different genotypes, so 6 different genotypes. In black are the ancestors. In blue are the evolved populations at 20 degrees. So even though they didn't find any new bits of trait space, you can see they did find new trait values. For example, this 1050 has much smaller cells than it used to have. And those cells have much less chlorophyll per unit volume than they used to. So this is evidence for cryptic phenotypes in multi-trait space. This just means that even though they're not moving around trait space per se, they are finding different ways of occupying that place. So how are they moving around trait space? Because if you recall, the populations that evolved at 24 degrees did move around trait space. Now, something about PCAs that is pretty annoying is that if you use all of your data points to make the PCA space, it shows you the greatest axis of variation, and it sort of hides the signal we're looking to see. So what we did was we went back to looking at all the pairwise correlations of traits when there was a correlation, and we made a correlation using only the ancestors. And so we have a correlation in 95% confidence intervals defined by the ancestors. We then projected all of the evolved populations onto that, and we gave them a score based on how far they deviated from the ancestral value. So populations where trait correlations did not evolve could still evolve their trait values by sliding up and down the ancestral correlation, but populations that evolved by breaking or making a new trait correlation had to leave this, and both their trait values and their trait correlations changed. They got an outlier score based on how far they were from the ancestral correlation. So the further away you got, the higher outlier score. We also summed that over all possible trait correlations. So the more often you did it, the higher your outlier score. Then we projected them onto a principal components analysis, or a traitscape that had been defined by the ancestors. The traitscape is remarkable because it explains over 90% of the variation in the data set. So this indicates that trait values in Thalassiosauria are actually constrained to very few major axes of variation. The ways to be Thalassiosauria are in fact fairly constrained. However, the individual trade-offs are not. If you look at the outlier score here, you can see that if you're moving right along principal component 1, so if your chlorophyll is going up, if your side scatter is going up, notice that growth doesn't change. You move along this principal component by deviating from the ancestral trait correlations, whereas you move in the opposite direction on that same principal component by sliding up and down the trait values while maintaining the ancestral correlations. Now, this pattern was really beautiful for the populations evolved at 24 degrees, but there was no pattern for the populations evolved back in the ancestral environment. And this is to be expected because this data has less variation in it anyway, right? They went back to the same spaces in trait space. So how are evolving populations moving around multi-trait space? How do we think of evolution when there are more than 2 traits involved? Well, if you're re-evolving in the ancestral environment, as might occur if there was some sort of colonization event, but you ended up in roughly the same type of place as you were before, the trait values evolve. We get lots of cryptic phenotypes, and the trait correlations evolve, but populations don't move that far from ancestral trait values. So while trait correlations evolve, they don't evolve much. In contrast, if you're evolving in a new environment, trait values evolve, you still get these cryptic phenotypes, but trait correlation evolution becomes incredibly important. So where are we now? I hope this was interesting. Where we are now is that trait variation does seem constrained. It in fact seems very constrained. 2 principal components axes explain about 90% of trait variation in Thalassiosauria. And we got about the same result when we did the the same analysis with 12 ancestors. So we think we have sampled the variation properly with our 6 ancestors. This may limit the ways to be a diatom beyond pairwise trade-offs, which is great. It means we can constrain the values of how diatoms are acting beyond just pairwise trade-offs. However, these pairwise trade-offs evolve incredibly easily, so we do want to move beyond them. And then the bit where there's still a lot of suspense is the actual traits. It looks like the trait correlations that evolve the most are the ones involving cell size and reactive oxygen, which is, I think, really interesting. We don't know what to make of that just yet. Why does this matter? Why have I told you all this? Why have I subjected you to principal components analysis? Well, first of all, diatoms and every other phytoplankton out there are not representable by single traits or pairs of traits. They're integrated organisms made up of many traits, and those traits are connected in ways that are higher order than pairwise correlations. So we need ways of looking at and thinking about multi-trait evolution that goes beyond pairwise trade-offs. Now, constraining phytoplankton traits in ecosystem models will make better projections about the future. But I'd like to note this, and I like to note it in red because it's super important. We don't need to know any of this to start petitioning hard to mitigate ocean acidification effects at all. This research is about projecting how weird things are going to get, which will help us inform how we lobby, but it's absolutely not new information that's needed to justify taking action. So I'd like to thank, with this, the team and our funders. This was funded by the Gordon and Betty Moore Foundation. This is the evolution and genomics team. Yes, there is genomics coming. That's being done at JCBI by Nate Walworth and Chris Dupont. The physiology team in Sydney was Martina Doblin and Phoebe Argyle. And the modeling team was Naomi Levine, Susanna Lelis, and Nate Walworth. You may notice Nate getting involved absolutely everywhere here. And finally, I have one or 2 minutes, and I just like to bring this up. I spend a lot of my professional life thinking about predicting what future phytoplankton will look like. But as you may be aware, there's been a pandemic, and we've all been stuck in our kitchens and living rooms and computers for a long time. And I got really tired of screens and started talking with a friend of mine on the phone with no screen, old school. And this friend just happens to be a science fiction writer. And we started thinking about what the future world we wanted to retire in would look like. And then we got an artist involved, and then we got an engineer involved. Then we got more scientists involved and a video editor. And what we have now is a collaborative illustrated storytelling project about life in future cities. We're calling it Choose Your Own Topia. The YouTube channel is launched. The website is going to launch in about 2 weeks. And what this is the story of the occupants of a building in Edinburgh in 2050. And there are 2 versions of Edinburgh. There's a utopia called Carrotborough and a dystopia called Edinstick. Yeah, and the goal is to try to involve people in other countries so that other people can tell the story of the future city that they would like to live in. So do check it out at some point. This is the team. We're made up of scientists, engineers, science fiction writers, artists, and film students, plus a few people who can only be described as feral kittlers, who just try to keep us all in line. All right, thank you.
Thank you, Sinead, for sharing so fantastic presentation. First, we are going to give time to some of our participants in this LAOCA course for asking some questions through our Zoom chat. And then Fernando is also capturing all the questions from our public chat on YouTube. So we have our first question by Victor Aguilera. He's from Chile and said, thanks, Sinead, very interesting talk. Ancestral diatom plus updates or new diatoms, how this might influence ecosystem processes?
So just, I think what you're asking is how the fact that diatoms travel, so the fact that in any part of the ocean you have a resident population and then a constant influx of immigrants, is that the question, how that affects things? The answer is we don't know, right? This is a great question. So if we decide that the current standing stock of variation in the ocean is all that's possible, then adding updates or adding immigrants is not going to change what's possible. It might speed it up, but it just means that fundamentally nothing new is possible, and it's just how fast you're making variation or mixing it. If you say that something innovation is likely, then it sort of has the potential to increase or decrease the frequency of innovation happening. But I'm completely not sure about that. This is something I'm working on with Ben Ward. We're really interested in how likely populations in the ocean are to adapt in situ in their current versus how likely they are to be replaced by populations that are migrating into their current. Because I've talked about this all like populations can be local in the ocean, but actually all phytoplankton populations are stuck in currents, right? And so they're traveling around in these wild fluctuating environments with some trading between currents of populations all the time, plus there's seed banks. So our model is very much a toy model.
Thank you. Fernando, I think there's already questions on the chat on YouTube.
All this without considering ecological interactions. Yeah, we haven't even gone there yet. The goal is to work with modelers who do ecosystem level stuff eventually. And so we're trying to get a handle on what's happening sort of within lineages. And then we collaborate with people. We collaborate to sort of throw that into models where we have more lineages and predation, right? Grazing is incredibly important for phytoplankton. So this is very much a bottom-up isolated bit that needs to be plugged into other models as well to get a sensible answer. What it tells us about is just the evolutionary processes. I'm over here. Hi.
Yeah. Thank you, Sinead. We have two questions from Juan Diego Gaitan. Fantastic talk, Sinead. Two questions. For evolved populations in novel environments, are these adaptive walks or more like sprint and then walk when acquiring losing correlations? That's the first question.
Adaptive sprints. Well, so in the model, we sort of, they end up being adaptive walks in the classical sense. In the experiments, we've not yet been able to get the resources to get a good timeline, right? So we sample the beginning, middle, and end of those experiments, and that's all we've had the power to do. And I would absolutely love to get my hands on a project that let me continuously monitor phenotypes over the course of an evolution experiment. So if anyone knows of a way to annex my own research institute so that I can do that, that would be great.
Yeah, we have a second question too. If evolved from Juan Diego Gaitan too, if evolved populations in novel environments return to ancestral environments, do they recover trait correlations from ancestral states?
No, they are more likely to, but not always. So what we saw in our experiment was that trait correlations evolved in both the back selection experiment at 20 degrees and when the populations evolved at 24 degrees. They just perhaps had more evolution of correlations at 24 degrees, but it was still rampant at 20 degrees. And rampant is the right word. It shocked us. This was an example of an evolution experiment that worked too well. We weren't expecting so much trait correlation evolution. And so when we went looking for it, it was like a data tsunami.
Thank you. There's another question from the participants. I am a bit confused with what you mean with changing evolving correlation between traits. Could you give us a practical theoretical example? Does it mean that the interaction between two traits would change with changing environmental conditions?
So it means that over time, the correlation between two traits could change substantially. So in our ancestral populations, so when we looked at the 12 ancestors, we saw, for example, the correlation we expected between size and growth, where the faster cells grew, divided more quickly than the smaller cells, which within Thalassiosauria was expected. Whereas after the evolution experiment, we had a lot of cells that were both small and dividing very quickly. And so they had escaped the usual correlation or the ancestral trait correlation. And this can be quite, to me, this was really interesting because a lot of times when we have models of how fast phytoplankton can grow, we actually will correct growth rate by cell size, right? When the environment changes. So if you're looking at trajectory through time, it looks like these correlations break down, at least within a genus, right? I'm not suggesting that suddenly cyanobacteria and the relationship in growth between cyanobacteria and like giant mixotrophs is going to change. We're looking at sort of within diatoms at this point. Counterintuitive, like I said, it was the data tsunami, and we remeasured many of the traits because we didn't believe them at first.
I have a question, Sinead. There's any antecedents, for instance, for mixotrophic organisms that can evolve by changing trophy, like from autotrophy to heterotrophy, or I mean, in the other direction?
Please do this experiment.
Okay.
I would love to know the answer to that, right? Because you would think that being a mixotroph would give you more flexibility. But as far as I haven't seen a lot of experiments with that. And having attempted an evolution experiment with Prorocentrum, with the dinoflagellate recently, I now understand why people very rarely do them. It was a mess.
Well, yeah, I can imagine.
I had a couple of questions too, if I can go for it. Sinead, looking at your data, one thing I find interesting is a pattern in from your ancestral line to the line that were off the peak of the fitness, and then to return different environment, there was a reduction in body size, or in size, sorry, I shouldn't say body, but in size, which I find interesting. It links also to what I'm going to talk a little about and no work you've done. Do you see this consistently through your experiment, that when you're put off your fitness peak or there's a reduction in size, and does this stay over multiple generation? If you know, or
I'm looking. Yeah, so we do consistently. So in experiments, we do consistently see size changes, but I wouldn't say we don't see it for all genotypes. And so it's something that seems to always have the possibility of happening, but it doesn't always happen. It no longer surprises me when it happens, we'll put it that way. And certainly reductions in cell size is something we see frequently in lab experiments with phytoplankton. And that sort of it's beginning to make sense, but I'm not quite there yet in figuring out.
Does it linger over generation? If you
it
continue to the selection, or?
Well, it's heritable, so if we put them back in the ancestral environment, they remain small.
Okay.
But it's always tricky with phytoplankton because you can't really it's hard to look at single cell division cycle resolution. We're doing like population measurements on the timescales of every few generations. But yes, it's heritable, and it does linger for a while.
That could be also an interesting link for your modeling with the ecosystem function question you had before, for productivity and energetics. And the other things that you hint to something quite interesting that I'm starting getting fascinated with, understanding nothing really yet. But you were saying before the correlations, they're not sufficient to describe systems, biological systems that are very complex. So are you telling us that the way forward, while we try to simplify things to constrain them and understand them intellectually, do you think that the way forward is complexity science for ecology and evolution?
I think we need to let it be complex enough so that we can find the simple patterns to put into models that can't represent process. So for example, the level of multi-trait correlations I'm describing in this study would be a disaster in a global oceans model, right? They're just computationally, it wouldn't work. But they would be doable in something like the model that Barton and Schaum and Henga did of trait evolution in different environments with phytoplankton where they were representing process. So my take on it is we need to let things get a little more complex when we're representing process in small models so that we can pull out patterns. And what would then go into the bigger models are those patterns. Bigger models don't need to represent all the underlying spaghetti, but we need to make sure we get the patterns right. If we put process in big models, that's an oversimplified process, we risk getting the wrong patterns. So I think there's sort of a multi-step nested model way that we have to go. I'm not advocating for complicating the Darwin model.
I appreciate both the question. The answer was really cool. This idea of complexifying, but also to constrain. And I also like the Italian connection with the spaghetti. So thank you for that.
Well, I mean, pasta solves a lot of things.
Thanks, Sinead. We have another question from the YouTube channel. Simone Baldanzi says, hello, Sinead, impressive results, and it was a mind-blowing presentation. And a question, can epigenetic mechanisms be involved in the reverse state, the walk back to ancestral, and how epigenetic mechanisms relate to trait correlation?
Ooh, so I think at this point, as a community, we've sort of gotten on board with the idea that epigenetic mechanisms are likely to always be part of evolution. So there aren't very many studies on how epigenetics interface with evolution. So if you look at some of the models that incorporate heritable epigenetic changes into adaptive walks explain the rate of evolution better than models that don't. So for instance, Ken Denman modeled the results that Kai Lobeck got with Emiliana huxleyi when he evolved it at high CO2, and found that he could only get the right rate of evolution. He could only get a model that reflected the actual experimental data if he allowed for epigenetic inheritance in the model, which is pretty cool. Do we know if it's involved with trait correlations? I don't see why not, but I don't think that we've looked at that specifically. But the answer these days, when someone asks me, could it be epigenetic? My answer is always yes, it could. It's not straightforward to check that. Yeah. So we've done experiments where we've sequenced the genome and the epigenome, and we don't find really clear correlations at that scale. We just know that the genome changes, the epigenome changes. There's really excellent work being done by Torsten Reusch on seagrasses and whatnot, if you're really interested in picking this one apart.
Thank you.
Okay, we'll see if there's any other question from our participants. So we're almost at time. Otherwise, Fernando, we can go on with the next speaker.
Yeah, we don't have more questions from the YouTube channel. I think Piero was asking for a few minutes. He will be right back. So maybe if you have any messages, well, we remind to our audience that all the questions are being received by the chat of the YouTube channel. So if you have any inquiries or questions, we're receiving them from there. And we'll have a quick break, a short break after the presentation of our next speaker today, Piero Calosi, and I think he's back now. So Christian, you may present him. Yeah.
Excellent. So, well, once again, thanks, Sinead, for such interesting and inspiring talk. We are almost perfect in terms of our time schedule. So we are going to present our next speaker, Dr. Piero Calosi. Piero is currently a professor at the University du Québec Rimouski campus in the Department of Biology, Chemistry, and Geography in Canada. His current research focus is the investigation of invertebrate physiological and life history response and the determination of their scope for further adaptation to multiple global environmental drivers, primarily ocean warming, ocean acidification, deoxygenation, and changes in salinity. Piero works from larvae to adults using laboratory natural selection experiments and in situ translocations to compare strains and populations in order to understand species' ability to adapt to rapid environmental changes. So today, Piero is going to give a talk with a polemic title, Size Matter, When the Small Dogs Wins. Thank you, Piero.
Thank you, Christian. The title has nothing to do with the fact that I'm not a particularly tall person, but let's see. So here, can you hear me? Can you see my screen?
Yeah, we can see.
Everything works?
Yep, perfect. Perfect.
So this talk was intended to talk about size during timeless changes, environmental changes, but also you'll see we'll deal with other matters in this respect. So the talk, in fact, will focus on body size, and in particular body size reductions, but also in rarity, which is something very important for biologists and a very, very intriguing concept in biodiversity. I start from rarity, this talk, going into body size a little later, because rarity is where I started my studies. At the University of Florence, where I'm from, I started working on this concept that geographical range size, you can see here. So these are species that have larger sizes of their range of distribution. These are species that are found in more restricted areas, and they're normally less abundant. Well, you have more species to be rare within a taxon, for example, fish or a group of birds or something, than species that are common, so find across a large extent of space. In particular, I worked on amphipods at the time, and I started dealing later on also with freshwater insects. So I abandoned the sea as a marine biologist for a couple of years during my postdoc in the UK. And my quest was to try to see whether there was a physiology of rarity, whether rare species compared to the common congeners have a different physiology. And the answer to that question was yes. I started looking at the mechanism that could define this physiological mechanism, in particular focusing on thermotolerance. And here I find in this really fascinating, extremely small, but really fascinating insect, freshwater insect, the thermal tolerant range correlated strongly to the extent of the range of a species or the central point. So the fact that we can find them more toward the north, the expansion, the post-glacial expansion. And the most fascinating part was that we could find the species that were more tolerant, on average, had greater ranges. They were also more plastic, like they had more plasticity. And species that were more endemics on the south, they were less tolerant and also less plastic. So this is making a double whammy, if you want. So there was a physiology of restricted widespread species, and this was the first time that was demonstrated in a large number of species using phylogenetic corrected analysis. So rarity make species more sensitive to climate changes. Apparently, that was our outcome. And there was a parallel that was made that I wasn't aware at the time by Barnowski in his Nature paper in 2011 of the fact that rarity is confirmed or was proposed from fossil record to be one of the traits that define species sensitivity to extinction. In fact, when we talk about extinction in particular, this is an example of the Permian Triassic massive extinction. When we have a massive extinction, we have normally a loss of 75% and more in biodiversity. We have arrived in some cases in marine ecosystem to 95, 90% of loss of biodiversity across this passage. Well, what we have a loss in biodiversity, but we have a loss in complexity. So we have phylogenetic and specific loss of biodiversity, but also in terms of sizes. So what we have observed that we have a loss of rare species and a preferential loss of large mass species. And this is quite intriguing. So what are the mechanisms? And this particular loss in sizes has been defined as a Lilliput hypothesis. I think all of you know the story of Lilliput, the traveling of this human sailor in country where there are giants as well as miniature people. And so the idea of nanification of ecosystem is something that is quite present and have a relatively good support, at least for marine benthic ecosystems in the paleo record. So what this hypothesis is saying that during time where we observe in the past warming, like we're observing now, ocean deoxygenation, like we're observing now, acidification of the ocean, like we're observing now, although it's important to say not within the same time scale, right? And changes being extremely rapid nowadays with global changes, we had a loss in biodiversity and a reduction in sizes of both the representative species that were left after an extinction, but also in some cases within the species, there was a reduction in sizes. Well, these of course make a parallel, an interesting but worrying parallel with the current changes in Anthropocene, where humans are the force of changes, both in terms of ecological change and also evolutionary pressure. And so we can expect that some of this pattern may be found. We also already are worried about reduction in biodiversity, and we have evidence that there might be an increase in species loss at this time in this period in time. This has been increasing, but still, whilst we have like certainty of the climate changes, we don't have as many certainty in terms of biological responses. The idea is that there's a lot of emerging properties, a lot of like surprises, evolutionary surprises that may expect, and therefore we still have like a very rough understanding of what could happen. So one of the reasons for this, as I said, emerging properties like biological systems are extremely complex. We have, for example, in marine organism, they have like a species that have like very complex life stages, sequences that we know still little. We don't understand yet quite well carryover effect, cumulative effect, aside for a bunch and handful of example of model species. We don't understand the function and the importance that existent adaptation, so local adaptation in particular, could play not just in modulating, but in making worse, the rapid impact the impact of rapid climate changes. And also the idea that we have, particularly in marine organism, aside for the work of some people like Sinead are doing, we still have a very little grasp of what transgenerational effect could have. And this both in mediating negative impact, but also in actually making negative impact emerge after a few generations, where if you do short-term experiment, we might not be able to be aware of. So the question I've had like for a while, it was whether we were under or overestimating biological impacts of ocean changes in biological system. And I had in mind always that, well, both. We might be in some cases underestimating the impact when we don't know that changes will happen over multiple generations. Or due to cumulative effect of carryover effect, they're negative in these cases. Or we might overestimating where we see negative effect that may be buffered later on. I think in general, we might be more underestimating this effect because we don't understand the evolutionary trajectory or the evolutionary space, and I make a parallel to Sinead talks, that could be left for some species to evolve into. So in order to try to make some sense and trying to make some generality emerge, if it was possible, with this colleague here, so Holly Putnam, Rhode Island University, Richard Twitchett at the Natural History Museum in London, and Fanny Vermandel, who is a PhD in my group, we come up with the idea to try to make a parallel in between among fossil records and patterns in changes in the fossil record, modern record for what exists in terms of pattern, the changes, and also like link it to physiological mechanism. Of course, we cannot make the physiology, we cannot study the physiology of fossils, but we can study the physiology of modern organism and trying to understand so what could have driven the past by using the future and using the past to inform what the future could hold. So what we did was, again, to concentrate in particular in marine metazoans, not just mass extinction, but like in particular mass extinction because of the most known study, the most striking also, but also looking at a general extinction that means like below 75%, but sometimes we talk about 20, 30, 40% loss in biodiversity. You can see here another example of more than an extinction event here on your left, where you have a loss in biodiversity, a loss in complexity of the ecosystem. Also, you have a smaller body size organism, less complex, with smaller, for example, tube that can inform also reduction in ecosystem function, which has been also indicated by fossil record, geological study, I should say. And then here we have a past extinction. Then we talk about after 1 to 2 million years, okay, to recover to a previous state of diversity where you have a much more complex ecosystem, many more species, borers, like that they make more complex and larger borers, so there's more oxygenation of the sediment with a greater impact, positive in this case, of biochemical cycles. You can see also the larger organism and more complex organism also re-emerge in a sense. And starting from this, we started trying to list all the pattern or response at the individual or at the geographical or the phylogenetic or the functional ecosystem level that we could think about and we could find evidence for in some cases not, but for the fossil record and the modern record. The check means that this pattern, for example, in this case of body size reduction, was observed in both the fossil and modern record. And we try then to link it to putative mechanism. They're normally like well investigated nowadays, or they've been indicated as possible mechanism that explain species sensitivity or tolerance to environmental changes. These are some and not all, so it's not comprehensive, but indicates some. One thing that clearly you can see, we have energy limitations, but this could be generally spoken as a metabolic limitation, energy budget limitation, also feeding limitation. And in particular, the two that I'm going to concentrate for this talk is body size reduction and loss of rare species. So for body size reduction, what we find is that there are like compelling evidence already for the fossil record and also in the modern record. We already have some evidence that point to this. And we were able to link it, at least according to theory, but also to actual evidence that provide a quantitative link to oxygen limitation. So if you want limitation to the physiological functioning under changes in marine organism, energy limitations here, and also biomineralization limitation. There could be more, but these are the links that seem to be the most solid and to link this. And what is interesting but also worrying that we also have, if you want, an interactive, synergistic appears in a negative sense, effect of selection in body size imposed by fishing, which is very well known, for example, by the example of cod in the St. Lawrence, where we had the cod fishery collapse and an impressive reduction in the maximum size the cod can reach, and the largest cod nowadays are a lot smaller than they used to be. So cumulative impact seems to be also the case to happen between direct anthropogenic effect in our level of activity, such as fishing, as well as the anthropogenic effect that we have caused through climate and global changes. So body size reduction is evident nowadays, and there's multiple pathways of impact in this sense that seem to indicate that this process could continue, could reach other species than those in which has been already described. These, of course, have important implication for productivity of ecosystems, okay? And the complexity also, the trophic chains, even within the intragill, for example, relationship, like you can find in some fishes where you can have cannibalism. We also have models that explain quite well how this happened, body size reduction. So this was a model that we produced for the paper, bringing together the paper from Sheridan and Bickford and also Algeo. More could be added, in fact, to this model that look at changes in CO2, in temperature, precipitation, oxygen, and many other processes might be actually due, for example, due to species interactions. So reduction, for example, in primary productivity, as we were talking about it with Sinead, with reduction in sizes in some microalgae or in some macroalgae, for example, it could happen, that have like energetic consequences. And that's an important aspect. We need to remember that body size reduction could be as easily explained by energetic constraints, where you have less productivity in a system. An organism that require a daily intake in energy that is higher will be non-favored, is not disfavored by that condition. So to give you an example, top predator normally, or even some top grazer, which are also predator, have normally large sizes. When there's less energy, less productivity in the system, those will be more easily impacted. And the reason is that while some organisms, for example, an elephant may have a metabolic rate, so consumption of oxygen, and so burning of energy per gram kilo, let's say, of biomass, that is lower than that of a mouse, the mouse will win, because despite consuming more for a gram of weight, is actually require a daily intake of energy that is much lower than that of an elephant. An example and a link that can be made in this sense is also that by the collapse of ecosystems in the North Sea, which is an area like a hot spot for climate changes, where what we have is the shift in Calanus, for example, due to climate changes, to species like more productive to less productive. So Calanus glacialis moving north and Calanus fimarchicus moving from south into the Northern Sea, occupying more area. Is that this actually was through modeling of ecosystem, it was detected as being the cause, or one of the primary cause of the collapse of some of the species, like, or reductions, or more than collapse, a major reduction in top predator, such as some of the birds like seagulls or sharks, and other macro species of the macrofauna. So there are a lot of evidence that demonstrate, first of all, that body size reduction is possible under climate changes, that is occurring, that could spread farther, and also that will have important consequences of a trophic change in ecosystem functioning. The other pattern I bring to your attention is the loss of rare species. So this is something that I started my talk with, the idea that rare species might be more sensitive during climate changes. This pattern was observed during fossil records, and in particular, when I talk about fossil record in this talk, but in general, the best known fossil record is for, in terms of ecosystem, is marine benthic ecosystem. When we talk about groups, I would say is probably foramins, foraminifera, and fish. They're really well known. But when we talk about ecosystem, is really benthic marine ecosystems, among all ecosystem confounded terrestrial, freshwater, and marine. In this case, for loss of rare species, what is considered as a putative mechanism, it could be energy limitations. So this idea to come back again, and physiological niche loss. In this case, considering also, we should consider that the physiological niche of rare species is considered to be already smaller, not just by the study I presented before from my own work, but by many other evidence provided for terrestrial, for freshwater and marine species. What we could also have considered, we didn't put it because we didn't have evidence for it, but we should investigate it, is whether rare species also have more oxygen limitation or physiological impairments, if you want, limitations related to the OCCP theory. And we don't really know this, whether there are rare species more sensitive to biomineralization limitation within the context of ocean acidification in particular. Other mechanisms could be brought to our attention. So we don't know whether rare species are more sensitive, for example, to changes in species interactions or other sorts of ecosystem changes. But another parallel is also the fact that because they live in more restricted area, so in a lower number of localities, and they normally also have already like a physiological niche is more restricted, it's reasonable to consider that they may be more sensitive to habitat degradation and loss. By chance, this is a common species or a widespread species, if you want to be more specific, have more chances to survive somewhere than a restricted species, rare species that is found only in few localities. So again, it comes the idea of climate change related and global change related, so physical-chemical changes might have an impact on rare species more than common species, but also direct human activity. So going back to the fact that changing our demography and changing our level of activity are our primary target for conservation, if you want to do so. But despite for this work, because that was a review that put together a lot of information, I'm also interested actually in running experiments, and it's a thing that I like the most. I like the most actually to experiment in situ where I can, but that's really hard and difficult to find global change related like natural situations and to make it easy for people. It's fun to dive and to snorkel and to do stuff, but it's also extremely demanding, and at times it can get dangerous if you're not careful, particularly working when you dive. So what I've been interested in the last few years, and it's something that we stop a second, like for a couple of years to take a breather, but we want to recontinue and we'll see at the end of this talk how. We are interested in seeing whether transgenerational exposure provided a level of protection to future combined ocean change drivers. So in particular, focusing on warming and acidification, but we're also moving toward working on hypoxic effects and salinity changes in coastal area, particularly as these are primary driver of changes in subarctic and Arctic condition with the melt of the snow or increased rainfall regimes, and also the melt, of course, of the ice cap unfortunately in our planet. In order to do this, we find the most convenient model or organism for us. I work on metazoans, so on multicellular animal organisms. And we find this really interesting for us, not as cute as a panda, of course, but really interesting model organism species. It's an annelid worm, a mobile species that can allow us to do multiple generation. Now, this is when it start getting more sticky, because we cannot do 900 generations, unfortunately. This is our limitation. So Sinead, I'm extremely jealous of your model and what you can achieve with those. So we can actually follow them in a lab quite easily. They don't have larval stages, which is a limitation if you want, because 25% of marine species don't have larval stages, but 75 tend to do. So of course, they're representative of marine organism, but to an extent, we need to consider this adding complexity of having a larval stages and more complex life cycle in most marine organisms. And with this was the team that actually undertook the work over a few years. They're now Some of them are still here, so Fanny and Maud, and are continuing their progression in the study, but the others are spread over the world doing different profession, working for UNESCO as a biologist, still continuing the work as biology, and becoming a conservation biologist, in this case, Cynthia. So what we did was that we decided to expose, to create some pairs and expose families to different conditions of control. You can see here in white, red warming, green we have acidified condition, and yellow we have combined conditions, and expose them over generations. So F1, F2, so create a sort of chronic exposure, but equally expose them just for a short amount of time, so through one generation to condition and switch to another one. We did a simplified experimental design when control was exposed to either control condition or all three experimental conditions, or warming, again, acidification are combined. And the experimental lineages, if you want, family, were exposed to their own ancestral condition or to the new condition, this being only the control. So we were all interested in comparing control against experimental against control, but now look what would happen whether we move, for example, a warming family into another condition, acidified or combined. What we measure, of course, time to first reproduction, number of reproductive events within a certain amount of time, fitness through number of eggs. We'll actually look also at egg sizes, growth of the embryo, which you can see here, this little blob here. And also we look at physiological proxies such as mitochondrial functions to see whether there were changes in the physiology. What we noticed, I'm not going to show you the result just for time issues, that we noticed that there was a decrease in some traits like growth rates of the embryos, of the larvae, sorry, and juveniles, apologize, reduction in maximum size or productivity in terms of fitness, like egg productions. But this was completely compensated in the second generation. So whilst we had a reduction in experimental condition against the control, once they were exposed for the second generation, they show actually to be able to compensate, and that's good news. So transgenerational exposure can be used as a way for a species to buffer negative conditions. However, we carried on this experiment for 7 generations. Each generation we did this sort of transplant laboratory experiment, trying to see whether we could see whether this ability to buffer completely a negative impact at the F2 was maintained over multiple generations, or maybe they could even improve in somehow and thrive under the new environments. However, under, in particular, like a warming condition, what we find is that there was a significant reduction in female size. And there was also a reduction in fecundity. Now, fecundity in annelids is dependent on the size of the organism, because every time they grow by adding new units, new metamers. So what are called catechers in this case, in this organism. And each catecher carries 2 new gonads. So a female that it grows larger or longer, if you want, will have multiple more gonads, and therefore a greater potential. However, even when we corrected for body size here to make sure that we accounted for gonad productivity potential, we noticed that there was still a decrease in fecundity. So we have both body size reductions that appear only after generation 5 and 6, so much later than what we normally do as experiments when we do 2-3 generation in metazoans. And we also notice fitness consequences. They're quite important, quite impressive. In fact, greater than female size reduction. This also makes the idea of the body size reduction a parallel with the Lilliput hypothesis. So the idea that there might be a link also in modern global changes to this hypothesis that was observed in the paleo record. What it was interested is that we noticed that when you look actually at the family we had created in general 12 pairs for these first experiments, but we have expanded this in time by going to 24 and 48 to have more solid data. Oops, and I see there's a mistake here. We had a number of pairs created, had to be increased under warming condition and combined condition to have at least 12 pairs, so to have comparable and balanced design during the F1 and F2. We also have a decrease, as you can see here, in success of hatching as percentage under these lineages. And we had appearance, particularly in combined, but also warming, and this started being really important in generation 5 in terms of number of extinction events. Please bear with me, there's a small mistake, well, there's a big mistake here, because this actually corresponds 6 pairs to around 50% of loss. And so we are on the edge of hard selection. Okay, And so hard selection and extinction might be what explain reduction in sizes. So there is a potentially preferential, both a preferential selection for small body size individuals, but also an evolution toward these traits. This hasn't been resolved, and it's just something I put forward as an hypothesis that we need to work on to actually to verify further. And with a lot more families and following both average size and maximum size very closely. So further to this, to provide some experimental evidence for these patterns in body size reduction, I also wanted to go back to my original question on rarity. And in order to do this, we actually selected 2 species of the same genus of Orchoca, Japonica diadema, which are 1 common and 1 rare species. Of course, please, and just for full disclosure, these are 2 species. So whatever I show you here is a provocation, is an intellectual exercise, more than a strong evidence. And this is a pattern that we expect to find everywhere. But it was the first evidence actually we wanted to put forward to also provoke in the community to start working on comparing rare and common species. So, and Cynthia Thibault did the same kind of experiment, but here we just limit, rather than do transplant, to chronic exposure. So to look what happened in F1 and F2 when family are exposed to control, warming, acidified, and combined conditions. So they're the same type of acclimation, the same type of processes, and the same also metric that were measured. I don't show you everything, but I show you the most, of course, interesting results. Again, we have a reduction in female maximum size, but this case only in the and this is, remember, F1, F2, only in the I lost the mouse, only in the rare species. There is this here represented with the circular symbols, while the triangle is a common species. You have here the reaction norm from control to acidified, control to warming, and control to combined. In this case, we see these changes occurring in the rare species across the warming and acidified. You can also see the intensity is really the same. So really, it's not acidification here driving these changes, but it's warming. And this interests every aspect of rare species, but as we expected, didn't impact the common species. Remember that the result I showed before, common species show body size reduction after generation 5, 6, while here the rare species show reduction in sizes after the first generation. So what it is that the rare species has a lesser ability for beneficial transgenerational buffering. And this was also observed for example, lifespan, which is quite important, of course, like for increasing risk of extinction or the risk of not being able to reproduce when conditions are favorable. What is also striking is that the rare species didn't make it to generation 2. So actually, we couldn't, under at least the warming and the combined warming acidification conditions. Again, extremely sensitive, and extinction in this case happened in the laboratory, even when we had a lot more lineages, 24 or 48, I cannot remember, but we are trying to increase like number of family. So apparently with extinction come then back, rare species are putatively more at risk of extinction, and also smaller species, both based on fossil record, modern record in ecological pattern ecosystems, and the presence in the literature of evidence for body size reduction. What I find most important for me is that whilst I know, for example, that colleague like William Chang was trying to include some evolutionary elements for metazoans or fishery importance. So some evolutionary elements in the biogeographical modeling. For me, it's also important that for creating generalities is to go and look at the molecular basis for this sensitivity. Because once we identify pathways or lack of pathways, molecular pathway, they may underpin the sensitivity, then it could be detected more easily across a lot of different organisms. And to do this, we are actually going in a dark side. I'm finding myself in a place where I didn't think I would have been nowadays, I have to say, in my career. And is that to actually start looking at cell physiology very closely using targeted and untargeted proteomic, lipidomic, and metabolomic approaches. I do it in collaboration particularly with Diana Madeira, who's a proteomic expert, and I started working with one of her colleagues, Rosario Dominguez, on lipidomics, while we can do metabolomic analysis and profiling in our university, our university. But I have to say that the bottleneck I find, and it's the most important, if you want a job, becoming bioinformaticians, is bioinformatic tools. The idea is that there's a lot of complexity in the data we generate. There's a lot of information that is not easy to also to bring together, to link. So you can characterize really well a lot of compartments, the transcriptome, the genome, the metabolome, the lipidome, the proteome. I'll continue a little, sorry, bear with me. But also whole organism responses, whether those are physiological, like thermal limits, CD max, CD means, metabolic rates, life history, growth, development, fitness. And that's the point, is to actually link molecular traits, molecular evolution to fitness. And to do this, you need incredibly developed bioinformatic tools. And the direction we are going in our lab, with the help of a very talented postdoc, Tessa Page, who is a American-British postdoc who is coming to join our lab in a few days, next week. She's arriving finally from Australia to the UK through all the pandemic and plane vaccinations plans and etc. But she's making it here to work with Diana, Carolina, Diana twin sister, as you can see, and Holly Putnam, to actually start developing approaches that can, in ecology and evolution, enable us to link all this compartment using a network of network approaches. What do I get now? My lab is full at the moment, but in the future, and I publicize this because there's very good students that come from South America and Central America, we are going toward reconnecting, if you want, the study of transgenerational plasticity and rapid adaptation with a very in-detail investigation of cell pathways. And this within the context of the physiology of marine invertebrates under global changes. So I actually we look for people, not now, but in a year time or so. So if you are interested, like to join the lab for the reason as a PhD student, but also as a postdoc and visiting postdoc, Quebec and Canada offer very good opportunities, also funding, having like scholarship for 4 to 1 year or 2 years for people that come and visit. So, and that person or whatever else would also join a very eclectic group of people that come from a lot of different backgrounds and countries, and have one common interest, which is to investigate the evolutionary physiology of marine organisms within the context of climate change, either using an ecological or molecular approach. And with that, thank you for following my rambling and my trying to connect different pieces with the purpose of trying to find some generalities if that can exist in these uncertain times. Thank you.
Thank you, Piero. Great talk. Thank you for sharing your time with the Latin American community. And I'm pretty sure your talk will be very useful, both in the framework of our current course on climate change evolution and for all the entire community connected through our YouTube channel. So
I see a question from Mauricio Urbino. I think I answered during the talk, but I can answer directly if you want.
Yeah, no, I mean, there are a couple of questions. I mean, we will try to consider both questions through the Zoom chat and through YouTube. And yeah, we can move on with the first question by Mauricio Urbina. It's a very long question, as you can see.
Yeah, but I think I answered, but I'll get again because it's quite important. It's something that I had in my mind for a while. Ciao, Mauricio. How are you doing? We know each other actually, so hello.
But maybe I can share the question for the community.
Go for it. Yeah.
In YouTube. So it said, "Hey everyone, Piero, nice talk. Due to metabolic scaling, it is a lot cheaper to sustain a gram of a big animal than a gram of a small one. The classical moose versus elephant. So miniaturization doesn't make sense in terms of metabolic efficiency, right?" Yes, it does make sense in terms of absolute amount of resource, as the elephant does use a lot more than a mouse. What do you think? Another alternative explanation is variability. A small animal deal quicker changes than big ones to environmental changes. So what do you think about variability shaping the decrease in size? You show us.
Okay, so I'll start again. Yeah, I agree with you that from metabolic scaling, from a I call efficiency perspective, it doesn't make sense, but actually it does, because metabolic efficiency, we normally physiology observe it on short time scale. But here we're talking a lifespan on which physiological ecologists normally don't work with, because our many organisms, like I work on lobsters, and it takes 5 years to get a reproductive lobster, and they can live for 60, 70 years. But the problem is that from a day-to-day basis, metabolic efficiency would explain to have larger organism is more convenient, because it costs less by gram. But on a lifespan, at the end of the day, you survive if you have enough energy, and not if you're more efficient. So an organism that require more energy would die more easily in a time where there's lower productivity. From a perspective of saying of variation, it depends again on the time scale. On a short time scale, when you're larger, actually, because your surface to volume ratio should protect you, defend you from negative impact of variation. But again, it's a matter of the time scale. And I think it is that paleontologists look at plasticity as something that happened over 10 of hundreds of thousands of years. We look in minutes or hours or days or weeks. And there is an issue here. We are not talking the same language, even when you use the same term. So I think we need to use a variable time scale according to the process that we are trying to define. So if you talk about extinction, it's not as the same scale that we talk about plasticity, which can occur in a very short amount of time. So on the case of variation of the environment, if I follow your question, I think it would favor small organism on a short scale, but it would favor big organism in a short scale, but in a long no, I'm sorry, I got it wrong. A small organism will be more impacted by variation. It can adjust more, but it can be more impacted, particularly when it's an ectotherm. So in this case, this is contradictory. But then what I think is in the longer scale, an energy requirement become more important. I think I got to the point of answering.
Yeah, thank you. Let's see. There are a couple of other questions on the chat from students. Okay, there's one from Isabel Timpe, a question for Pietro, not for Piero, for Pietro. Will these compensation effects from the exposure to traits may be shown in generations of other more complex organisms, corals, for instance? Can you see?
Yeah, I'm not sure I understand the compensation. Yeah. So we are talking, I guess, about buffering, so compensating through transgenerational exposure. Effect from exposure to trait may be shown in generation of more complex organism. I'm not sure I follow. I'm not sure I understand completely the question. If the question is related to complex organism, if they can show negative effect in short amount of time, it's possible. Often, for example, it depends. I'll try to answer this question. Corals, it depends whether you consider it more complex than worms. I teach invertebrate. I can tell you that worms are more complex from a tissue organism or organs organization than anemones and corals. So they're actually more simple. For example, they don't have mesodermic tissues. They don't have a complete intestine. So worms are more complex. If I take, for example, the cephalopods, like a squid, in that case, the physiology is to push the limit of efficiency and performance that actually are demonstrated to be more sensitive to environmental changes that go outside their actual current ability to buffer conditions because of this complexity and the fact to live at the limits. So yes, we could expect to have complex organism to show negative impact over generation in a short amount of time because they already live in a way like I make squid as an example. I'll tell you why. Already at the edge of their ability to perform so highly functions. In fact, the oxygen limitation hypothesis was first postulated in 1936 by a German scientist who called it the asphyxiation hypothesis, because at every cycle or circulation, a squid already have to extract all the oxygen for the bloodstream in order to perform this highly functional, narrow function, locomotory function. So if you just push them a little farther into more temperature, oxygen, or other stressors, they will show negative impact more easily. And this could be considered to happen more faster in a multi-generational sense. I hope I understood the question correctly.
Thank you. Another question, after mass extinctions, do you think that the species evolved to optimize access to resources in the new scenario? And that implies optimizing metabolic processes?
That's a really cool question. If you want to come and answer in our lab, do an experiment, would be great. I don't have an answer for that. There's no way that there are, to actually answer you in terms of the fossil record, because unfortunately, fossil physiology, the physiology of that animal doesn't exist. Otherwise, we'd have the answer. I think, again, is an issue of scale. Remember that to reconstitute an ecosystem, it takes 1 to 2 million years. And that's the amount of time it takes for a species to emerge, 2 million years, like more or less. So Optimizing metabolic processes. I think that it's possible that following right after an extinction or during a time where there's the collapse of a trophic chain, the energy available is so low, in general is considered to be so low, that for sure you would benefit for having a lower metabolism. So optimizing it to lower metabolic rates, Natalia. Because, well, lower metabolic rates, I would say, sorry, lower metabolic requirements, going back to my idea of it's a maximal energy at the end that matter. And in fact, as an example, an organism, like a multicellular organism that does really well after extinction are mushrooms, because there's a lot of organic matter decomposing and they don't have major metabolic requirements, right? A high level of metabolism. Second question was, do you think that species life cycle and reproductive strategy could become simplified in response to changing environmental conditions? We published a paper in Scientific Report with Noel Lucy, who is a research fellow now at the Smithsonian Research Institute in Panama, in Boca del Toro, where we find that in the CO2 vents in Ischia, but also in a lot of like human impacted and extreme conditions, the species that seem to do best in terms of marine benthic organism are those that don't have larval life stages and they have modified life stages where the maternal cares, maternal investment. And it's something that some had put forward some time ago, actually, so a few years before that we published the paper, where we had actually done experiment in the vents, not saying that there was a species exclusion, and all the species inside the CO2 vent were with direct development, modified development, and the one outside, they were more dominant with larval development. So yes, I think that a simplification would be the case in terms of like reduction of life stages and also maternal investment or parental investment. Why does salinity affect the size of top predators? I don't have an answer for that question. Actually, I don't know. I never studied that, or I know there's evidence that salinity may do that. But if you do, we can discuss it in a separate maybe venue.
Thanks. Then another question by Valesca San Martin. So smaller species plus low reproduction, results in rot to extinction. Is there evidence in marine mollusks exposed to multiple stressors, these being the ones that have been shown to be the group most affected, by example, by OA, temperature, or deoxygenation?
I really appreciate you really have like a lot of complex and interesting questions. That's a great challenge. Thank you, Valesca. Well, it's a rotful. To be a small species actually is in a way seem to be an advantage. So small species are the one they're going to resist or persist, sorry, I should say, across extinction more likely. That's what the fossil record tells us. We also have evidence, modern evidence in terms of like observations, surveys, that there is a reduction in the size of some species under climate changes and global changes, where global, remember, include habitat destructions and fishing, overfishing, targeted fishing. So the road to persist extinction is to be small and not to be rare, to be a common species, if you want to persist. In marine mollusks exposed to multiple stressors, I don't know. So there is some evidence, and actually something from a personal observation, that it hasn't been quantified. So that's important to remember that we are in an age of quantitative science. But in the oxygen chronic environment that we have in St. Lawrence, at 300 meters, we have area stations that are chronically hypoxic. We notice a reduction in biodiversity, and of those species that persist in this hypoxic zone, we have a reduction in sizes. On the top of my head, I don't remember of evidence on the climate changes or reduction in sizes in mollusks in general, but it could be sensible as a hypothesis. And I don't remember. I'm sorry. I should know this actually. Because also in the fossil record is what we find. So a lot we find bivalves that persisted over extinction. They are the smallest species, and within the species, the smallest individuals, or they evolved to be smaller and developed to be smaller. So I would expect that under similar condition in worming, hypoxia, and acidification, the mollusks in general would reduce in size. As I said, we see here this in terms of like the bivalve in St. Lawrence, as I know there is evidence on the fossil record.
Right. Carlos Vergara Chen from Panama. He asked, in marine invertebrates, metabolism and respiration increases with increases in temperature and CO2 along with a reduction in seawater pH. Does this mean that populations evolving in stable environments will be the first affected by changes in the ocean before populations evolving in highly variable environments?
I partly agree, Carlos, what you said about the fact that under increasing temperature, metabolism tends to increase until you reach a point of, a breaking point. It can also lead to death, like the Arrhenius point, for example, when the heart functions start failing or other functions. On the CO2 is more debatable because we have every possible example. We have example that with CO2, metabolism increases, stay the same or decreases among different species, but even within the same species among different populations. I show this in Littorina littorea, where northern population, central population, or southern population of Littorina littorea in Europe respond differently. I think there are evidence that already, and this idea has been put forward a few years ago, that also by, for example, a review in 2008 by Spicer and Widdicombe, and has been tested further, the organism from stable environments may be more subjective if you have local adaptation in general, or they live in more stable niches to be more sensitive to actually to global warming. With Sarah Magozzi in Plymouth, we actually published a paper comparing shrimp species from all the same decapod group. 3 species that were from stable environments and 3 from more variable environment, where we find that one that come from variable environment are more equipped to deal with the changes itself, and we look at it. But then, I'll be contradictory here, but then in terms of like the long term, the chronic conditions, they actually have too high energy expenditure. So yes, in general, I would say this species from stable environment seem to be more sensitive. But again, we have never looked this into a multi-generation context. So we might have the surprise that while they're better equipped physiological to deal with these conditions, they may not be energetically convenient as life forms. So I said to you something, and then I contradicted because it's part of biology having a lot of emerging properties. The idea, yes, is species from more stable environments seem to be more vulnerable, but this should be tested in the context of a multi-generation experiment, I think, before to actually put the final, full stop to that sentence.
Okay. I have another question also from Carlos. Is in tropical regions where environmental stability is assumed, organisms exposed for hundreds of thousands of years to seasonal changes due to coastal upwelling could have transmitted environmental tolerance from generation to generation, with new generations that could possess a greater adaptive potential than organisms inhabiting environments without upwellings?
That's really cool. I'm struggling here, but they're really good questions to everybody. I really enjoy it, so thank you, Carlos. It's interesting because I'm going for my sabbatical, I'll be in Chile, so I'll come and cook with Christian. That's my plan for my sabbatical. We're going to make tortellini together, I decided. But I'll be also in Panama, and I wanted to go and see the upwelling areas there. They're really intriguing habitats. So again, I'll be rambling because I don't have a straight answer. It's not something I thought through very well yet. If you come from an upwelling environment, you're used actually to variable environment. So in a way, you should be able to deal with variation better. Of course, as a maximum condition increase, you might find yourself that in dealing with conditions that you're not used to, so you have less tolerant than species that come from not upwelling area. Okay, so I agree with you that if tolerance to heat is heritable and is within the range of your tolerance, then the species that come from non-upwelling area should be more tolerant and could pass this to the progeny. For those that come from non-upwelling area, there might be they had to trade off the ability to deal with variation with the ability to tolerate a certain maximum level. And there is evidence sometimes for trade-off in these traits. Like I didn't find them in the diving beetles, which however seem to be more of an exception than a norm in some cases. So I think that the idea is both yes, when you come from a variable environment, you might better equip in variation, but then less for these maxima. They're particularly important in the tropic, particularly I think in the tropic because they're really high temperature, and we can get close to sort of molecular malfunctioning to get to the maximum of the ability of molecule to evolve to tolerate like really high temperatures. So I think it's going to be an interplay between irritability of dealing with variation or dealing with maxima and where the maxima are. Because of course, even if you're able to deal with really high maxima, but that goes above your ability, then not. It might be the species from non-upwelling area. They're actually not equipped to live there because the environmental maxima surpass their molecular stability. So in a way that the safety margin is not functional anymore. So species, and then in the case species for upwelling area may be favored by the fact that where they live, they have a break during the year that can tolerate, they can recover from negative impact or season of not upwelling where you don't have any break in environment. Carlos, I hope I answered correctly your question, which is really cool, but I never thought in this way, and it's really interesting. So well done for challenging me here.
Yeah, we'll have the final questions. I mean, Simon Baldanzi, your Italian colleague in YouTube, he asked, "Ciao Piero, nice talk. Rarity and abundance are linked to ecological interactions too. Would you suggest to add biotic interactions on top of environmental stressors in experiments? Have you done that?"
Ciao Simone. We don't know. We know each other from internet, for emails and Zoom, but we never met actually with Simone. So I hope to meet him one day. No, of course. I'm whilst I'm a physiological ecologist, I'm well aware that like species interaction drives a lot of changes in species distribution, abundance, locally and globally. In fact, there are evidence from a paper that was published in Nature Climate Change on the fact that in an initial phase of an extinction, and this we're talking about past extinctions, the loss of biodiversity is due to directive impact. So the chemical and physics of the environment will drive local and global extinction of species. But after this, when you start degrading the complexity and the redundancy in some functions or trophic position of some species, well, then it's the interaction that changes, because it's not the direction of interaction that change. A predator will be a predator and a prey, but it's the intensity of this. And of course, if you're a predator and you don't have any more prey, well, you're gonna die. And this is an impact of an indirect impact, okay? Absolutely, it's very important. I must admit that it's extremely complex to test this in control environments, and to do this also ideally through generations when you deal with metazoan, I think is best suited to work on yeast and bacteria and macroalgae to ask this kind of questions, or in microbial community in general. We have tried in the past to come up with some ideas, and unfortunately weren't funded. But it's something that we have tried in the past, and it require extremely large teams that have like very complementary expertise and a lot of time, because it takes years to, I think, come up with solid experiments and sufficient replications, also repetition of experiment, to make sure that what we see is not spurious results, but like solid scientific evidence, right? Consistent patterns. So very cool. Yes, I could be able to do it, but I think macrobiologists are a better place to do this actually. And I've done it in the past already, like some work on, I think Sinead, also she worked on extinction in the past.
Mm-hmm. Just a quick question. The last one. Typically, evolution experiments at the lab keeps animal up on constant conditions in 2-3 different treatments for instance. How this static condition could affect results, especially when we expose organisms at the laboratory to variability regimes different than the experience at field. I know that it's a big challenge. Does it matter in evolutionary experiments?
Yes, immensely. I mean, we need to understand that the laboratory is an artificial environment, is an artifact that we create, and we create the artifact to answer the question. So, of course, I agree with you that in a lot of cases, the result would be different if we had natural variation. And studying variation, I mean, there are some experiments out also. Sam did some work on this, and on a cnidarian, if I remember correctly, you're going to have different results, or the result that differ because of the condition. Absolutely. So I think we need to understand that a lot of the work that we do sometimes are proof of concepts that we put forward to inform big picture, because it's virtually impossible, at least on organisms that are complex in particular and have long life cycles, to be able to do this work easily. I mean, 7th generation was 1 year of work and almost the death of 2 students. So we have to be realistic about what is the scope and the breadth of our results, to recognize the limitations. And indeed, when we can go and test it outside, or even better, there's a really interesting chapter for you guys on a book on experimental evolution by Garland and Rose. And there's a chapter written by Ray Huey, which is one of the, I think, my most favorite, if you want, scholars on Earth. And it's called Catch-22. It's an old movie from the 1950s or 60s on the nuclear war dilemma. No, when you have nuclear war weapons, so it's dangerous. When you use them, are you going to be the first or the second? But doesn't matter, because if you're the first or the second, everybody's going to die nonetheless. So there's a situation that turn around like a cat biting its tails. And he says, experimental evolution is important, that you do it in the lab to actually fix things and to study in controlled conditions so you can dissect the factors and the drivers. But then if you don't see it happening in nature, it doesn't make any sense. So you need to work both in nature along gradients or in alternative conditions as well in the lab. And the two go together. You cannot discriminate them because in nature, you cannot really tear apart the factors and say, yes, this is happening because warming is causing this or acidification is causing this. I think that where possible, and a lot of this, we don't use as modern biologists fossil records sufficiently to actually even, put, if you want, so you do experiments, you do field observation, but if you put a third leg, the table will stand actually, will be sufficient. And so we need to do this triangulation among different tools. A fourth, which a fourth leg to the table that Sinead pointed is modeling. And modeling can be done projecting things and then testing them, or actually to do it on existing data to actually reconstruct a model. So I think the idea is to use different evidence, or triangulate as you do in geometry architecture to try to find really the pattern that seem to be, you can almost say is the truth. Truth doesn't exist in science, but let's say that is almost true. Okay.
Okay, thank you very much, Piero and Sinead. I will give the pass to Fernando. We're going for a short break. And but please keep on in case you will need to answer the question to Sam.
Yes, thank you, Christian. Thank you, Piero, of course, for his great presentation. Also Sinead. Actually, Sinead said, for good overviews of variability in evolution experiments, see Donadiela experiments by Luis Caviel Chevin. He gets it very systematically so that we could actually take types of variation into account. We will have then, right now, we invite you to a short break of 10 minutes. So please stay connected to the Zoom meeting and the YouTube transmission. We will be back soon at this LAOCA webinar, Ocean Acidification and Climate Change, Perspectives from Evolutionary Ecology. We will have then our final presentation and speaker, so stay tuned. We are back here in 10 minutes. Thank you. Thank you all, and welcome back to our webinar, or LAOCA webinar, Ocean Acidification and Climate Change, Perspectives from Evolutionary Ecology, organized from the Latin American Ocean Acidification Network, the Coastal Social Ecological Millennium Institute, SECOS, and the Millennium Institute of Oceanography, IMO. We welcome back all of you who are following us from the YouTube transmission, and we also remind you that we will receiving questions from the YouTube chat. So if you have any inquiries or questions, please let us know, and we will be reading from there. Christian?
Thank you. Thank you all the audience for keeping on in our third LAOCA webinar, this time with a very special issue in the framework of our LAOCA training workshops, ocean acidification and other stressors associated with climate change, a look from evolutionary ecology. Our third and last speaker is Dr. Sam Dupont. Sam is a researcher and a professor in marine ecophysiologist at the University of Gothenburg and an honorary assistant professor at the School of Biological Science in Hong Kong University. Sam's main research topic is focused on the effect of increased CO2 and related changes on marine species and ecosystem. His works aim at revealing the mechanisms behind a species and ecosystem response to environmental changes and developing the needed unifying theory for large-scale projection. So Sam is in direct contact with many stakeholders, both at the local and global level. He's a member of the Advisory Board of Ocean Acidification International Coordination Center, OICC, and the Executive Council of the Global Ocean Acidification Observatory Network, GOAON. So Sam is actually doing a research state at the International Atomic Energy Agency in Monaco, and he's at this moment flying back to Sweden or in the middle of an airport terminal somewhere. So, but the good news is that he sent us a video with his presentation. Hopefully he will be able to answer all your questions through the Zoom chat and your YouTube channel at the end of the presentation. So in case he didn't have any delay with the flights. So otherwise, he said his very good friend Piero would respond all the questions. No, joking. So well, let's go to the final talk by Dr. Sam Dupont, entitled A Question of Time. How much do we need to understand evolution to address and minimize ocean acidification? Thank you, Sam. If you watch it on YouTube later.
Thank you, Christian. If you can, please let me know if you're hearing the presentation of Sam. I will play it right now.
I think you need to put the audio.
I will reproduce it again because we have a slide.
You need to open, probably Fernando, you need to open your microphone and then turn on.
Yeah, please let me know if you hear it now.
Hello, everybody.
Yeah.
This is Sam here.
It's okay.
Okay.
Nice
Okay. Thank you.
Have the chance to present today about the work we've been doing on the impact of ocean acidification with a focus on time. So I'm sorry I'm not here in person at the moment, but I'm actually traveling back home. I'm working half-time these days in Monaco, so I'm recording this from the IAEA and the Ocean Acidification International Coordination Center. But I hope that if my plane is on time, I will be able to join for the questions afterwards. But to be on the safe side, we decided to record this. So let's start. So I guess you heard a lot about climate change and ocean acidification and evolutionary perspective over the last days. So I won't give you any introduction and immediately go into the core of the presentation. And I'm sure that that's been a big part of what you talked about over the last days, the fact that over time, there were in the field of ocean acidification in particular, more and more studies highlighting the fact that we had huge differences in sensitivity between species, and even more puzzling between populations of the same species. So this is one of the papers from Christian showing that, for example, two different species, the population of the same species of copepod along the coast of Chile were responding completely differently to the same scenarios of acidification, and one was responding really positively, the other one was responding negatively. So at the time, that was really annoying, this kind of data, because that means you can't extrapolate from one species to another or to one population to the other because you don't understand what's happening. So we were definitely missing something. And basically one of the pieces of the puzzle was the fact that people were ignoring the huge natural variability that you have in different places. And that's something that could explain, for example, the difference between the two populations of copepod here. One was in a more stable environment, and then over time, through evolution, adapted to these conditions and were much more sensitive than the other population who was living in a more fluctuating environment. Definitely, that highlighted the importance of taking into account local conditions and local variability if you want to understand species sensitivity to a stressor like ocean acidification. And I'm pretty sure you've seen that paper many times over the last days, where Christian and other colleagues re-evaluated the literature and found this beautiful index allowing to take into account the local variability and re-evaluate the literature, and actually explain why some populations are more sensitive than others. So local adaptation and variability, as you know very well by now, is critical. But there are some other gaps in the field of ocean acidification, and that has been nicely summarized in 2015 by Ulf Ribesel and Jean-Pierre Gattuso. So that's another one of these iconic figures. But basically what they were saying is that the science we need is completely different than the science we have today in the field of ocean acidification. We most of the time work on single species and using experiment on one driver at a time over a short period of time, where what we want to understand ultimately is what's going to happen over a long period of time in a complex multi-stressor environment and at the ecosystem level. So basically the point was, we need to work more at the ecological level, we need to work on multiple stressors, and we have to consider time. And what I'm going to talk about today is really this axis, like time, and how time can play a really important modulating role, allowing, for example, evolutionary processes to occur. So time is impacting, for example, the chemistry of the ocean at different scales. So we are talking about acidification, which is a long-term change in pH. So slowly things are going down in terms of pH. But you have other levels, other scales of variability with time. So for example, you have variability that is very predictable. You have this cycle between night and days, and we're going to talk more about that. So that's extremely predictable, and organisms can adapt to that. And you have more unpredictable events. So you can have extreme events like heat waves, but you also have the equivalent for ocean acidification. You can have, for example, upwelling events where the pH will drop significantly over a period of time. And then that can also play a role in how we design experiment, because the duration of exposure to the stress can be different. So if you expose for 12 hours, for days, for months, for years, that can have very different consequences. So we might actually over or underestimate the impact of acidification in experiments if we don't pay attention to the role of time. And time could be on an adult. You can expose an adult for a long period of time, but you can also consider trans life cycle, so multiple life stages, one after the other, or even through generations. So some of the questions we're going to try to answer today during the presentation is like, are we actually using appropriate experimental designs, and what are the limitations, but also what is the modulating role of evolution. So let's start with the first part of the variability, which is this short-term predictable variability. So I will present some preliminary data for a lot of experiments we've done in my lab. Some of them are still under analysis, some others were published, but it's going to be like 6 different stories, 6 different little experiments illustrating different parts of what I just talked about. So basically how time can modulate the response of organisms to ocean acidification. So the first one is short-term variability. And a good example of that would be the day-night variability created by photosynthesis. So this work is something that we've been doing together with Damboya Kosa. So she's one of my PhD students, based mostly in Mozambique. So she's really interested in seagrass, and particular seagrass restoration. So we were interested in the consequence of seagrass restoration for marine calcifiers, because if you have seagrass, you're going to have more variability in pH, meaning that might have a consequence for organisms living there and calcifying, because basically, even if overall the average pH might be the same, the minimum pH that the organism experience will be lower. So that might actually impact calcifiers. And so we basically designed an experiment that you can see on the right-hand side with a lot of aquariums that were fed with water with 4 different pH, so 8.2 to 7.5. And then in half of them, we had seagrass, and the other half, we didn't have seagrass. And we, of course, replicated all our aquariums. And then we used sea urchin larvae as a model for calcification, because it's very easy to measure calcification in those guys and growth. So the data will show you now are the preliminary analysis for calcification and growth, but we are still working on all that. But basically, it's important to restore seagrass. That's something that is really big because it's a carbon sink. It's also played a lot of the other role in the ecosystem. So that was the background for this. But then, as a side effect, you have a lot of variability. So during the day, you have light and photosynthesis and respiration, but overall, you have production of oxygen and capture of carbon dioxide, meaning that the pH goes up. During the night, it's the other way around. You don't have light, you don't have photosynthesis, you only have respiration, and then pH goes down. So the question was, how will organisms respond to this variability? And we, of course, documented the chemistry, and it's an example from one aquarium. You can see in blue the pH in one of the control aquarium without seagrass, and the pH was fairly constant. You have a little bit of variability, but that's okay. And then you can see in orange, like the cycle of day and night, where as predicted, the pH goes down during the night and goes up during the day. And overall, when you check all the aquarium, adding seagrass really increases the variability. You have 4 to 8 times more variability in presence of the seagrass. Then we were checking a little bit like what is the consequence of this variability. And the first thing we could check is the net calcification over one day. So we just checked how much they were calcified every day, and then you can compare the different pH and if there is a role of the presence or the absence of the seagrass. And overall, there is this trend that is classic, that if you decrease the pH, you also decrease the calcification. So nothing really new here. But interestingly enough, there was no difference when you check the net calcification between the aquarium with just the water or the seagrass. So when you check overall for the net calcification, you don't have much of a difference. But then if you check at a different time scale, so instead of checking the calcification over one day, but you check like the difference between day and night, so we measure calcification every 12 hours, then you have something like that. And that's what you see in seagrass. So you have the calcification rate through time. And what you can see is that you have this really nice cycle where they calcify much more during the day, where the conditions of the water are better, like you have higher pH. And then during the night, they don't calcify, or they even dissolve sometime during the night. So you have this really low calcification rate, and what is below 0, that means it's dissolving. So the skeleton is dissolving. And when you check what's happening in aquarium with just the water, then you still have some kind of cycle, but it's less predictable, and it's kind of getting a little bit erratic at the end. So it seems that even if there was no variability in the aquarium, the larvae were anticipating the variability. And by doing that, we're still following a little bit of the cycle, but that doesn't synchronize as well as in the presence of the seagrass. So we have this cycle of calcification, but it's much stronger and much more regular in presence of the seagrass creating the variability. And then if you look a little bit closer to what's happening in the different pH, we have something like that. And that's where we see that there is a huge difference between the behavior of the larvae in seawater without the seagrass or with the seagrass. So on the left-hand side, you can compare what's happening during the day and during the night. So during the day, you can see that the orange dots, that basically the calcification rate is constant during the day and really high, and it's not impacted by pH. So it seems that when the conditions are the most propice for calcification, they just try to maintain the calcification as high as possible. While during the night, actually, they don't do that. So you can see that there is even dissolution. So they basically put all their energy in calcifying during the day where the conditions are better, and then they shut it down during the night. While if you check when there is no seagrass, when the pH is more constant, in that case, there is no difference between day and night, and they have basically this relationship between calcification rate and the pH. So you have two different environments, a constant and a variable, and two different strategies in how the larvae respond to pH. And so in one case, they invest all their energy during the night where the conditions are better, and they avoid calcifying during the day, and they avoid calcifying during the night when the pH is much lower. And in the other case, it calcify always at the same rate, so no effect of day and night. So two different strategies, two different phenotypes. The next question was like, is there a difference in fitness between these two phenotypes, or are they doing the same or not, depending on how they actually cope with this variability? And to do that, one of the parameters we checked is growth. And when you compare growth between the similar kind of average pH, 7.5, 7.7, and so on, in presence or absence of seagrass, you can see that you have the classic trends that the lower the pH, the lower the growth rate. So nothing new there again. But interestingly enough, in the lower pH, 7.7 and 7.5, you have a decrease in growth rates in presence of seagrass. So that means that they have a different ways of coping with the calcification. So they calcify more during the day, and less, and not at all during the night, and even dissolve during the night. And that has a cost, because probably they have to invest a lot of energy in compensating for the loss. And that can be shown here on the growth at the lower pH. So there is a decreased growth with decreasing pH and an extra cost of being exposed to variability. And I won't show you that now, but it seems that the parameter explaining the best these differences between presence of seagrass or not is the minimum pH. So that's another argument in defense of the index that I mentioned before, is that even in experiments that we do in the lab where we create variability, minimum pH seems to be the main driver. So short-term variability has an impact. You have different strategies in copepods. So they don't do calcification in the same way when they are exposed in variable environment or constant environment. To go one step further, we did another experiment where we had a much more controlled variability. So that was kind of a pilot. We used sea urchin again, another species though, Strongylocentrotis rebbachiensis. And what we did is we kept the larvae into either constant or fluctuating environment. So that's what you can see on the little graph here for 10 days, allowing them to be selected. So they die during the process. Some of them die every day. And after that, we either kept them in the same condition or we transferred them to the other condition. So we transplanted them. So some of them spend all their life in fluctuating, some of them spend all their life in constant, and some of them were transferred from one environment to the other. And again, we are processing those data, but what I'm going to show you is the impact on the survival or the mortality rate. So if you keep them all their life in fluctuating environment or in stable environment, they basically have the same mortality rate, and there is no difference in the first phase of the experiment and the second phase of the experiment. So they basically from stable environment to fluctuating, you can imagine that it's a stress or to be exposed to a more variable environment. So it's not surprising that actually you have an increase in mortality when they are transferring to fluctuating environment. But surprisingly, you have the same trend when you move them from fluctuating to stable. So you have this increase in mortality when you move from fluctuating to stable environment. So that's typically probably a cost of being plastic, cost of plasticity, where when you move from stable to fluctuating, you have cost of canalization. So you basically have indication from these two experiments that I just mentioned that you have different phenotypes out there in the population of the sea urchins, one that is better at coping with fluctuating and one that is better at coping with stable environment. We don't really know yet if it's really a selection process or if there is something else like an acclimation process, but we did an experiment this spring to test that. So we did similar kind of experiment, but it's a bit more complex. And we also played a little bit with inverting the cycles and so on. And we collected data also for genetic analysis. So we're going to see if we can associate these different phenotypes with different genotypes and see if it's a selection process. So that will be for next year or another time we can see each other. But overall, we have different phenotypes linked in larvae that are exposed to constant and fluctuating with different strategies to do the same thing. And there is a cost if you take this phenotype and transfer it to an environment where it's not being selected. So that's for the short-term predictable variability. Now you can ask yourself what's happening if it's more unpredictable, and how long is too much, basically. If you expose like a larvae to abruptly to a new condition that they never experienced before, how do they respond to that? So basically an acute exposure, and then how long before you have a negative effect? And that's an experiment that we just finished 2 weeks ago with one of my students, Medina from Germany. So for that, we use brittle stars larvae, and we basically compare two different pH. So we expose like the larvae to either a high pH or a low pH that is deviating from the present range of natural variability, so a real stress. And then we expose them for different durations from 1, 2, 3, 4, 5, and so on, 30 days. And then after that, transfer them back into their control water and checked what's happening for their survival and their growth. And it's really, really preliminary. I just checked the data yesterday for the first time, but this is what we have so far. And basically, it seems to show that for both growth rate and mortality, you have a relationship between the duration of exposure and the response of the different parameters. So what you can say is that the more time you spend at low pH, the more negatively you're impacted for growth rate and relative mortality. So even a couple of days that you spend in suboptimal conditions has carryover for the later stage, even if you go back to your normal conditions. So there is a cost even for 1, 2, or 3 days spent in low pH conditions. So this shows the importance of these extreme events, and that it can have, even if it's really short, can have cascading effects. So you have this negative carryover effect even of short-term exposure. Then you can ask yourself, okay, now we talked about a few days or daily variability. What about longer term? What about weeks, months, years? And for that, I will talk about an experiment we did, like a 4th experiment that we did on sea urchins several years ago. That one is published, it's been published in 2013. And for that, we just tried to go one step further and not just do the acute exposure. We just collected the sea urchins, acclimated them to the lab, and then kept them in the lab for 16 months under 2 different pHs, like 8.1 and 7.7. And then we designed a follow-up experiment to test for carryover effects to the next life stages, which were larvae and juveniles. But for the first part, we checked like what is the impact of long-term exposure on the adults, and that was kind of cool because after 4 months, we checked the fecundity of the adults. So like after 4 months exposure to low pH, and what we could see is that you had a strong negative effect after 4 months. But then if you kept the experiment running for 1 more year, then we could not see any effect. So this effect disappeared after 60 months. And so we interpret that as the time needed for acclimation is quite high. And that's something that has been documented in sea urchins before, where you could see, like, for example, if you transplant them from one environment to the other, they have the ability of reshaping their skeleton, but it takes 6 months. And probably when you transfer them to low pH, what's also happening is that they have to reshape their physiological mechanisms. So they have, for example, to have different types of pumps or different quantities of pumps, and that might take quite a lot of time and cost also some energy. And the energy to do that is probably taken from the gonads, because it's well known that the gonads in sea urchins are used as an energy reserve. So what we saw, this strong decrease in egg production after 4 months of exposure was probably a transient cost to acclimate to the new conditions. But then when we checked the carryover effect, what we did is basically compare the response of the larvae to high and low pH. And we also played with the acclimation of the adults before. So first, for that, we checked how many juveniles were produced at the end of the experiment. And so if we just collected the sea urchins that were kept for the 16 months at controlled pH and then test the impact on the larvae, what we could see is basically there was no strong effect of the pH on the number of juveniles produced. So there's same amount of juveniles in larvae raised at high and low pH. If we did the same experiment with the adults kept at low pH and then checked the impact of high or low pH on the larvae, it was the same. There was no effect. But what was a really strong effect was basically comparing the acclimation of the adult. So if you acclimate the adult at high pH, you have much higher number of juveniles than if you kept them at low pH. So you have this strong negative carryover effect from the acclimation of the adults to the larval stages and the production of juveniles. So that shows that in that case, the direct impact of pH on the larvae was not that strong, but there was a really strong carryover effect from the exposure to the adults. So in conclusion, for that part, when you do longer term experiment, you can have very different results than when you do it in the short term. Even if you focus on one life stages, the adults in our case, you can overestimate the effect, because in that case, we could see that the negative effect observed was just a transient one and it disappears later. Or you can underestimate the effect, because you could see there was no direct effect of the pH on the larval stages, but a really strong negative effect when you were considering both the exposure of the adults and the exposure of the larvae. So you have strong negative carryover effects here. And then if we keep thinking about increasing the duration of our experiment, then you can start doing really cool stuff like transgenerational experiments. And then you can start to focus really on evolutionary processes. So I have multiple generations. Now I have two examples for that. The first one is studies we've done with two cool dudes, Peter Thor and Pierre de Witt in Sweden. So we worked on the copepods, Pseudocalanus. And this experiment, the idea was really to see the transgenerational effect. So we had 3 different pHs that were selected because the high one is the average pH that the copepod is experiencing. 7.75 is the extreme of the natural variability. So still something they experience today. And 7.54 is a really true OA scenario that is outside of the natural range of variability. So this work has been published in 2 different papers in 2015 and 2016. And the design was quite complex. So basically we collected copepods in the field and exposed them to the 3 different pH for 2 generations. And after that, they were transferred to different pH conditions for 3 weeks. So either maintaining the same pH all their life or transplanted to another one. And the idea really was to also try to identify direct and transgenerational effects. So what is called original population in the future graphs are the population of copepods that were kept all their life in the same pH, so 8.1, 7.7, or 7.5. And the transplant were the one that were acutely transplanted from the pH they were kept in for 2 generations and then transplanted for 3 weeks in a completely different one. And you could see that we didn't do all the cross for practical reasons. But the story is quite nice at the end of the day, because this is one of the main graphs that we had. So first, if you check just what we were calling like this original population, if you just compare the pH effects when they were kept all their life in the same pH, is something like that, like acutely exposed, to the different pH, you have something like that. So basically the acute effect of low pH, so if you keep them at 8.1 for 2 generations and then transfer them acutely to low pH for 3 weeks, what you have is this. You have basically the higher the PCO2 or the lower the pH, the more negatively impacted they are. And we checked in that case a number of X per female. So there is a negative effect of low pH of the acute exposure. That's something we knew and we've seen in many of the other experiments I mentioned in this presentation. But then what was interesting is to compare this to the one that were kept all their life in the low pH. And that's what you can see, for example, here, if you compare for the 7.75 treatment, you have a completely reversible thing. So it doesn't matter in that case if you keep them for 2 generations at low pH or if you acutely expose them to low pH. They have the same response and they go back and forth. So in that case, it's a good example of plasticity. And that makes sense because they are exposed to such conditions naturally. So they are just really good at coping with it. But then if you expose them to conditions that are much more drastic, much more negative, a real stress, as you can see that if you acutely expose them to these conditions, it's actually very negative impact. So the number of X per female decrease significantly. But then if you keep them all their life at low pH, then you have a compensation effect. You can see that they actually have much less negative effects. So you have a transgenerational effect that is minimizing the negative effect of acidification. So when we saw that, we couldn't really know if it was a selection process or an acclimation process, like a transgenerational acclimation. So to make the difference, we collected samples and did RNA sequencing, and that's the other paper. And what is really cool about that paper is the pipeline of analysis. So at the end, when you do RNA sequencing, you end up with a lot of data. So we had more than 200,000 contigs, and we could do gene expression, of course, but we also identified more than 700 SNPs. So we could really try to see if we could find specific pathways that are over or underexpressed, or if we could trace selection processes. And by what was really cool was the fact that we used physiological hypotheses. So what we saw in the previous graph I showed you, to select the genes that were behaving in the same way. And you can have a look at the paper if you're interested, but basically we have really strong arguments there to say that this is a selection process. So this transgenerational effect is really through selection. So we selected some genotypes that are better at coping with low pH. And then we could also identify some genes or some pathways that are actually potentially selected, and it was RNA transcription, ribosomal function, ion transport, and oxidative phosphorylation. So that was really interesting to see that even after 2 generations, you're able to select for genotypes that are better at coping with low pH and then compensate for this acute negative effect. So stress, in that case, can totally lead to positive selection in that species of copepods. So 2-3 generations. And the final experiment I will present is even longer. So that's a crazy experiment that I designed with my colleague Saif in Kuwait. So they have really amazing facilities there. So we went nuts and we designed this crazy experiment that was done. And we just had the data, part of the data a couple of weeks ago. But basically, we compared the response of 2 different species of copepods. And we exposed them to 9 different temperatures, covering the present and the future range of variability of temperature. And then we had 3 different pH regimes. So we basically compared either like a high pH, 8.2, or a low pH. So you abruptly expose the copepods to this low pH of 7.5. And then we had also a slow rate of change. So basically you decrease the pH a little bit at every generation. And the reason we did that, because that's what's going to happen in the context of acidification, is like a slow change over time. It's not this abrupt change that we often do in the lab. So we wanted to see if this pH regime had an impact and compare if there is like if it's better or worse to have gradual changes, and you have arguments for that it could be good or bad. So we had 3 pH regime, and then we kept the copepods for 13 generations. So that was basically more than a year experiment and a lot of work. And for the preliminary analysis I will show you today, I will just talk about egg productions. So this is a summary graph of the kind of pH response you had at the different temperatures. So if you just check at the temperature first, it's the classic temperature curve. So the number of eggs is increasing with temperature till you reach a threshold, and then it's going down. So that's nothing really new or exciting. What was fun was to actually compare the pH response. So if you acutely decrease the pH to 7.5, you actually have this shift in the performance curve. That's classically what was predicted by Hans Pertner's model. So in a way, it fits really well in that case. But what was kind of also interesting is that when you compare the gradual pH change and the acute pH change, then you have very different responses. With the gradual change, it's much less negatively impacted. So it's more or less the same curve that you have with the high pH. So even if they reach the same pH in the gradual and the acute one, the gradual changes was much less negatives. So we wanted to understand a little bit why. So for that, we also analyzed how the egg production was changing through the different generations. And most of the time we had something like that. So basically the number of eggs was changing, either increasing or decreasing through time. So that's an example of an increase where you can see the number of eggs increase from 3 to more or less 6 over the 13 generations. So we could calculate a rate of change. So how many more eggs or less eggs they are producing through time. And when you put this number in relation with the initial number of eggs after the first generation, so you check how many eggs per female were produced, and then you check how fast they were producing or changing the number of eggs per female through generations, you have this amazing relationship. And I think that's really exciting. I was really excited when I saw that, because what you can see there is that there is a kind of an optimal number of eggs, which is about 12, 13 eggs here on that axis. And if they produce that number to start with, they just don't change. They keep producing the same amount of eggs. Then if they produce less than that, they start to actually produce more and more eggs per generation. So in that case, let's say you produce only 5, but the next generation, you're going to produce a little bit more, and then a little bit more, and a little bit more, till optimally you want to reach these 12 eggs per female. And then if you produce more than that, because the temperature is higher, you actually start to produce less eggs. So you try to reduce the number of eggs you produce through time, so you can reach, again, this optimal egg production. So the rate of change through generation depends on how far you deviate from the optimal 12x condition. And interestingly enough, in that case, there was no difference between the different treatments. So the pH or even if it's gradual or acute, it didn't make any difference. And also remarkably, it's exactly the same between the two species of copepods. So that's fun. So you could use this kind of data to make a little bit of basic modeling and see how long it would take to actually compensate for a cost. So let's say that you're in the condition where it's 20 degrees. And at the beginning, if you check at the high pH, it produces about the 12 eggs per female, which is the optimal condition. But then if you acutely decrease the pH to 7.5, then the pH, the number of eggs is reduced to 7. And then you can check by using this rate that I just showed you how many generations you need to go back to normal. And it's more than 40 generations. So we can estimate how long it would take for the copepods to actually go back to optimal conditions. So what is really cool here is that we can see that we have the potential for adaptation here. So the copepods are able through time to actually compensate and go back to optimal conditions. So stress can lead to selection, and also what was interesting in that last experiment is to show that if you have a slow rate of change of the pH through time, so not these acute things, they seem to have enough time to actually compensate very quickly, and then you don't have such a negative effect. So in terms of OA for these guys, this is a good news. That means that slow rate of change will allow the copepod to adjust quickly enough. So in conclusion of all this, and I hope you enjoy the trip, is that if you really want to understand what's going to happen in the future, you have to consider time, and you have to consider short-term changes. I showed you that even like daily fluctuations can lead to different strategies for a larvae to cope with low pH. The duration of exposure is very important. Like you can over or underestimate the impact if you too short. The variability in itself can also drive change. We could see that the larvae were selected to have phenotypes that are better at coping with variability. And then the rate of change is very important. If you have gradual change versus acute changes, you can have very, very different responses. So all that have impact on processes such as acclimation, carryover, selection, and all that can modulate the biological response. So that's really, really important to think about that when you design your experiment or interpret your results. Sometimes you can't really do the experiment you want. We all have limitations, but at least you have to think about it when you interpret. So thank you very much for listening to this video, and I hope that I'm here now, that the planes were on time, and I'm able to join you for a little discussion. Hello, everybody. This is Sam here.
Okay. Okay, so unfortunately, Sam apparently couldn't connect, but since we have some time, maybe we can open the opportunity for discussion and open question to the speakers which are connected, Piero or Sinead, so we can follow anyway questions on the chat, both on Zoom or YouTube, in case you have any additional comments or questions. Yeah.
Sinead and Piero, maybe you can turn on your camera so we can have you here also. Oh, you're there. Thank you.
I have a question for Piero that I didn't get to ask because you were.
Please don't push me. Oh, no, I thought it was on some presentation. Okay, so you were pushing me under the bus already. Okay.
I was just really curious because you're using you're using a lot of paleontology or fossil records to combine with modern studies about size evolution. And I've always been a little worried that the data I see on size evolution are an artifact of working with asexual microbes, right? So in my experiments, I never let the microbes have sex because I'm mean. And that means that there's always the possibility, especially with diatoms, that they're just getting smaller because they're asexual. And I don't think that's entirely the case because I don't see that in the controls, and the controls are also not having sex. But I've got to thinking that a lot of the size selection on being big is because of trophic interactions, right? So if you want to eat someone or avoid being eaten, it's better to be big. And we notice this a lot with phytoplankton. They like do all sorts of stuff to appear big by making spines and whatnot. And grazers are gape limited usually. So being big is great, but if there's some sort of catastrophic event, rather than being limited by trophic interactions, you suddenly become limited by energy, and then it's much better to be small. So do you think this, like, there's just a shift, depending on how fast the environment is changing, there could just be a shift in selection to be big from trophic interactions versus selection to be small from energy? And in the lab,
would you say make much sense to me?
Never have trophic interactions, right? So in the lab, we just keep on giving those energetic selections without any counteracting trophic selection.
What would you say makes much sense? And you sort of join me in the idea that we look things at the time scale or within a context, but then within the context of an extinction or of a cascade, an extinction cascade, because it's kind of a cascade also, the extinction, that then the process and the selection will change. So it is possible that in the short term of the life of a species to be bigger, like larvae in the zooplankton do the same thing. There's sometimes they come out of an egg, like small crabs, and they have this huge long spine. They're like 7 times their body length. They're actually extendable appendices that become rigid within minutes, in seconds actually, to just be bigger so the fish cannot gobble them. But then if they're really expensive energetically, then why keep it? Or you'll be selected against in a time of a disaster of energy flowing. So yeah, it makes absolute sense. But I do think that these questions, and I understand your caveats in using asexual and sexual reproduction. I know how complex it can be. Like I can imagine. This is best addressed in community of unicellular organism or very small organism where it's possible to create community. Although I think it would be best to test it on artificial community, like where you actually, as you can do in the lab, like compose your own artificial community and test the idea more than trying to complexify it. But no, you're right. I think it could make sense. And yeah. And it's interesting that you say that you don't see this reduction in control, but you see them in the selected lines.
Well, we don't see it as much in the control. When we bring things into the lab, they often sort of get a bit smaller, but we can usually explain that because we keep them asexually, right? So, I mean, I have the benefit of I can do experiments that are 900 generations, but of course the bad thing about working with phytoplankton, or the limit, is that we have limited life history control, right? So we know that they form resting spores and we know they have sex, and we have very little control over that for most of the things we can study in the lab. Whereas you guys have this life history that you can study.
Yeah, and that is our limitation. The life history can be very complex. Right? So I just wanted to, I don't know if there's any question that me and Sinead wanted to discuss, but one thing I find interesting in some data, and I'll tell him that he could try that when he was showing the last experiment, that he just received the data, so maybe it's elaborated. He fitted some curves, and which is interesting, but like they were, I think, hand fit, and he'll do analysis. In that case, it looks like the threshold of response don't change much. While in the theory of tipping points, when you add an extra stressor, an extra situation, you expect the threshold to arrive earlier, to be outpaced. I think in English the right verb is. Well, I didn't notice that. Of course, I don't have a quantitative eyes to say, okay, this is calculation, but it's interesting. What it was interesting was a difference in the intensity response when you add the acidification stressors, which is really cool. Although I have to say that I disagree with Sam that I didn't see a difference in the response between low and rapid. When you look at the points, like we don't know what is the variation around it, but I didn't see anything so strikingly different. Is the presence of acidification and seem to add this extra stress that makes the response more intense. But just in case for all the participants, like I've realized by working on scenarios, and it's something that we do, and it's normal because we simplify experimental design, that they're really useless for policy making and for management. That the thing we should work on, as someone was showing in gradient of temperature, is actually working on gradients, and maybe it makes a cocktail of a gradient of your primary drivers with adding the scenario of the second driver. So this is a really good thing, because In that way, you can then have the information to develop, to put those data in models and translate them into modeling exercises that are useful for management, environmental managers, and policymakers. We've done this with some as an exercise with the Southern Californian Ocean Current Water Project, or something that worked from the government of California, that tried to develop bills based on indicator on threshold of pH. Of course, it can be done in temperature, salinity, anything. But so remember in your career to develop eventually gradient approaches, which seem to be useful, both theoretically for proper modeling, could be evolutionary, ecological modeling, biogeographical modeling, but also because it is more readily translatable into policy and management.
Your microphone, Shin, is
there we go. That better. Yeah. I would echo that. I mean, Madril, Thomas, and I are submitting a paper tonight, hopefully, actually talking about the types of questions that can only be addressed with gradients. And the starting point for the paper that we wrote is that we reviewed the past 15 years of studies of phytoplankton looking at the joint effects of temperature and ocean acidification. And after 15 years, we still can't make the interaction surface. And that's ridiculous. These are things you can culture in the lab where we can do gradient studies, and they're not being done because they're demanding. And so we did point out that using only scenarios, you can't detect a lot of tipping points because driver interactions are not linear. And sort of also by using only scenarios, you can't tell how they're not linear or how the interactions are not linear. So it gets very dodgy. Anyway, I'm expecting a lot of pushback on that article. People are not going to like it because we're saying we have to do bigger experiments. So that's definitely, I think, key when you're looking for thresholds in multiple dimensions, is that eventually we have to get away from the ANOVA type studies. ANOVA type studies are a great place to start, and they're often a necessary place to start. But for most organisms, they can't be where we stop.
So, Sinead, if I can just launch you like a provocation, my students are done threshold on heat waves gradients, and there's a package what was called segmented on R that was used classically to do tipping points, but you use, of course, linear relationship and look when it's not linear anymore. But there's a new package that uses a Bayesian approach. And the difference is while you still look for linearization of processes, you actually can find multiple points. And sometimes you can tell them, look, there's a point around here, there's a point. So you can actually indicate, and it's a lot more flexible as a tool. It's a lot more complex, so we don't yet understand it completely. But they're going toward this idea of understanding the biological reaction. Often they're not linearizable easily, but that you can have actually multiple segmentations. And that could be something that you might.
Yeah, but we need to put data into these for
Yeah.
organisms. I mean, so we do have the statistical and like modeling capability to deal with multiple gradients with more than 3 gradients interacting, but until we know about the equations that let them interact, all we're doing is guessing at those from first principles, and our knowledge of physiology isn't sufficient to generate an interaction equation from first principles at this point, right? So it's not a question of whether or not we can construct the models, but whether or not we can parameterize them. And I think that's where the experiments are lagging behind. It's part of being like in evolutionary biology, you're always sort of about 20 years behind the theory, running to catch up with your experiments, I find. Well, I say you, I mean me. We are always so far behind the theory trying to put numbers on it. And then sometimes you just find out that bits of the theory, some of the assumptions that seemed obvious were either wrong or not generalizable. And that can be quite dangerous if you're building policy on it, I think. But I don't want to stop on a negative note. So I hope someone else has a question that will let us, well, it's not negative, right? It's saying do more experiments.
Yeah, but I think it's sorry, we're going on a tangent here, Christian, and sorry guys, but it's also the problem, like more and more we do partner research where we actually listen to the stakeholders, and I think there are good times where science should always be strong and solid theoretically, but also have an open ear to what are the needs, so that you can still answer your theoretical question and do fundamental science, but it can also be more useful. And fundamental science is always useful because more knowledge or basic knowledge is useful. but it can be designed in a way that answer also other needs. And I think that that's important, like from fishing community, for example, but up to policymakers. But you need to know before you start. As you say, sometimes you look back 15 years and you realize that you don't have the good data, which is shocking. Right? I say, what have we done in the last 2 decades? And of course, what it was done, it was great, and it was what it was needed. But retrospectively, say, okay, maybe we can do more and better, and always.
Yeah, well, I feel like I can publish this paper because I'm the worst offender at these in terms of these designs. I started doing them, and when we started, it was what was possible, and now we need to move beyond that. But I think you do have to listen to stakeholders. But sometimes, if we're being asked for thresholds, sometimes we need to put our foot down and say, well, we can only calculate the threshold with this if we can generate these equations, and we can't generate these equations without these big experiments. And so I think sometimes you do have to walk stakeholders through what sort of basic science is needed to address the question that they need addressed. And it's not always as straightforward as everyone would like. Sometimes you have to be like, well, we don't even have the theory to do that yet. And we can make the theory, but it's going to require some work. So I think we need to make sure we're going in the right direction all the time. I think we're agreeing on this. If we had Sam here, he'd find a way to agree with us as well, but sound like he was disagreeing. This is what Sam and I do best. Do we have questions, comments? Does anyone else want to jump in?
Very cool discussion. Well, very cool discussion and reflections at the end of the webinar. There are some questions in the chat for Sam, but maybe we can send them to Sam later by email. So we don't want to expose our guest speaker to question for other speakers. So, well, thank you very much for your time, for your enthusiasm to collaborate with these activities of our Latin American network. I'm sure it has been very beneficial for all of us and the entire community. So thanks a lot. Maybe Fernando, we are going to close the session.
Thank you, Christian. First of all, we would like to thank, of course, to Sinead Collins and Piero Calosi, who will be with us today. Sam Dupont also for his presentation. We hope to have him in a future webinar. Thank you to Christian Vargas for being with us today too. We also remind you that this webinar is being recorded and it will be available in the LAOCA YouTube channel. So if you can check it out later, you can see it there at the LAOCA YouTube channel. Thank you all, of course, all the students from the course of LAOCA. Thank you all for participating in the third Latin American Ocean Acidification Network webinar. And follow us for more activities through the Millennium Institute in Oceanography, IMO, and the Coastal Social Ecological Millennium Institute, SECOS, social networks and websites. We will be informing for further activities of the LAOCA networks. Thank you very much and see you in the next LAOCA webinar.
Thank you. Okay.
Thank you all.
Thank you. Bye-bye.
Bye. Thank you.